Laboratory of Oncology Institute, Rotary Cancer Hospital, All India Institute of Medical Sciences, New Delhi, and Professor R.K. Kale, ex-Vice Chancellor, Gujarat Central University, and Faculty of School of Life Sciences, Jawaharlal Nehru University, New Delhi. Dr. Ritu Gupta is a professor and officer in charge of Laboratory Oncology Unit at AIIMS and Laboratory Services at National Cancer Institute, Jhajjar. She is also the Vice President of Cytometry Society, India. She is the member of the Central Core Research Facility Committee of AIIMS. Professor Gupta leads as Principal Investigator of Unit of Excellence on Multiple Myeloma and Center of Advanced Research and Education on Acute Myeloid Leukemia. She has her major research interest in hematological malignancies. Professor R.K. Kale has his research expertise in oxidative stress, biochemistry, analytical chemistry, biotechnology, and molecular biology. He has received many awards in his research career. The recent ones are Maulana Azad Puraskar for Educational Upliftment in 2012 and Lifetime Achievement Award given by Dr. Baba Saheb Ambedkar, Marathwada University, Aurangabad in 2015. I request the honorable chairpersons to please take hold of the session.
Yeah, thank you very much for our nice introduction. Professor Rituji, shall I start? Hello?
Yeah, Ritu, you are muted. She's saying yes.
Yes, okay.
So please go ahead. There is a lot of background at my end, so I've
okay. Yeah. Okay. Thank you so nice of you with your permission. And at the outset, I would like to express my grateful thanks to Vivaji and his and friends for giving an opportunity to attend this conference and also chair this honor, indeed honor and privilege. Today, we have this will be the 7th lecture in biological dosimetry for radiation exposure, and I think Professor Dr. Dwarkanath will speak on radiation-induced cytogenetic change, chromosomal aberrations, macronuclear formation, and mutation assay. There is no need of formal or informal introduction for Dr. Dwarkanath. I think it will, if you do informally also, it will overkill effect like in India, what we find in radiation biology. So I will request him to give his lecture, and I hope everybody will enjoy this one. And he's a wonderful speaker. I still remember the first lecture I heard he has given when we were very young, actually, at that time we were beginners, and I heard him at Jaipur University. I still remember his lecture. So Dwarkanath ji, this is a floor for you.
Thank you. Thank you, Alessa, and thanks Professor Ramesh Chandra and Akhilesh Verma to be also amidst us. It's a great pleasure always. I have a couple of things as a prelude before I discuss a little bit about the cytogenetic damage and the biodosimetry for radiation exposure. We had yesterday broken off in cellular radiobiological effects lecture 2, since some discussion of that needs to be done, followed by, to the extent possible, I think since we cannot have too much spillover, I will probably shrink the cytogenetic damage and its utilization as a biodosimeter for radiation exposure, so as to fit into the time limit, which is an hour and a quarter. So to begin with, let me complete with the permission of the chair and also Dr. Viba, the things that were left from the last lecture. And I also want to confess, due to several compulsions, Dr. Viba had to give me this responsibility of covering the dosimetry, but that's not my forte, as all of you know. But I'll try to do a job as good as I can. And I'll be happy, of course, to interact later to clarify things. Due to paucity of time, as also the vast field of biodosimetry, I might probably either run a little faster at time, or pay attention to only a few things which are not contained in the slides. All right, then let me just go where I left yesterday. And
Dr. Dwarkanath, you can go up to 11:30, then we can discuss if you have anything.
oh, 11:30, yeah, because it's now 10 and it's 10:12. That's why I saw it.
So you can go up to 11:30 and the discussion can be combined with the tutorials.
Okay.
If you want,
So,
if you
yeah,
want. Otherwise,
yeah, even then I think, sure. Yeah, that's so
Yeah,
nice.
that's okay. That's okay. I think that
Yeah.
will work.
Good. So we left here yesterday. Let me share the slide. Okay. Share. Okay. Now. Why?
you were on slide number 35 or 36 somewhere.
Yeah.
No.
Is it on?
Yeah, it's showing.
It is showing. Why am I not able to see?
You have pinned it, I think.
No, Vibaji has some secret this one.
No. Let me see.
So
Why?
she keep
why
the
is
record.
it?
You the record.
Wait a
It's
minute.
there. It's there actually. 35.
I
Maybe
know. I
My,
also,
my,
no, yeah, it should be fine. Okay, is that now okay?
yeah. We can
Alright.
see radiation in.
Okay, this is where we left yesterday. We were discussing cellular radio response of the several things that we discussed, starting from the dose response for survival, various biophysical models, pre-molecular array, post-molecular array, PLDR, SLDR. We completed everything. And we were at a place where we need to discuss the relationship between various cellular responses to outcome, particularly with a focus on radiation-induced cell death. And I had stopped here summarizing that radiation induces both DNA as well as non-DNA damages that elicits two very broad group of responses, one that drives the cell towards survival, the other one towards death. And included a couple of things here which are missing in this. Autophagy is one of them, which plays a role both in a context-dependent manner, regulating or pushing cells to survival under certain conditions and to death when incomplete autophagy takes place or abortive autophagy takes place. So the consequences when the cell goes through a hierarchically regulated signaling pathways initiated following the lesions, both DNA as well as non-DNA damage, the outcome or the consequence, final consequences could be the loss of clonogenicity, either due to a mitotic death, which is closely linked to the cytogenetic damage, as well as both primary interface death, that is apoptosis, necroptosis, as well as secondary apoptosis, which ensues in the post-mitotic cells. The pre-mitotic cells are the early apoptosis. All this contribute to the cell death. And what is also important is the induction of senescence and stem phenotype, which have consequences to the efficacy of radiotherapy from amongst those tumor cells that were not killed. Those that are killed, no matter how or through which pathway, we don't have to worry discussing what else is the phenotype they acquired. Those which do not get killed, they survive. And if they acquired due to the exposure to radiation, altered phenotype that makes the tumor more malignant, including ability to invade outside its milieu, as well as metastase to be able to locate at other parts of the organism or humans, and the stem phenotype, which is responsible for when the stem cell is recruited back, they're more aggressive, which is synonymous with the epithelial to mesenchymal transformation that occurs, responsible for regrowth of tumors, both within the volume irradiated, as well as probably disease dissemination. So these are the consequences. I will probably discuss with the DNA damage repair to the extent that is permissible. As I said, probably I'll shrink to a very large extent. Now, failure to repair damage can either lead to death or mutations in survivors. And that's one of the reasons as to why one would like to study, amongst other things, the repair of DNA damage, as well as damage related responses like cell cycle perturbation, the checkpoint activation, the interface death, the apoptosis, as well as necroptosis, so on and so forth. The basic tenets of repair of DNA damage can be summarized as 4 Rs. This is not to be confused with the 4 or 5 Rs of radiotherapy, which are very different, that we will, I'm sure it has already been discussed by Dr. Giridhar, but we can also discuss when we will talk about radiosensitization. So irrespective of the type of lesions that are formed that we took account of yesterday, which are different types, the base damages, offload of base damages, base loss, crosslinks in the DNA, both intra and intrastrand crosslinks, and crosslinking with the protein associated with the DNA, both histone and non-histone proteins, DNA protein crosslinks, and most importantly than anything else, the strand breaks, and particularly the double strand breaks, responsible for the cell death even at moderate doses. So while DNA repair encompasses the repair of all these class of lesions, from the viewpoint of IR-induced DNA damage repair, you will see much of the focus, and therefore the existing literature dwells on the repair of double-strand breaks, and we have a great deal of insight, but much needs to be understood. Now, for having said that, let me just give an overview that the 4 Rs consist of, firstly, irrespective of the lesion, the damage needs to be recognized. That's actually the sensing of damage, the existing machinery. We'll come to as we see a couple of repair pathways. Then responding to the lesion for having recognized and activating several other related pathways, which includes probably the checkpoint activation. If the lesions are such, maybe death activation through P53 independent and dependent pathways to apoptosis. And this is also parallel to the repair of lesions, successful repair of lesions, so that the genome can be restored, integrity can be restored, resulting in resetting of the checkpoint. So the DNA damage sensor leads to the generation of several signals, as we will go in the next few slides, precisely for different types of lesions. But as a class of regulators, we can say sensors, mediators, effectors, and executors. I'll talk about the base excision repair very briefly. Base excision repair comes into picture when a base is damaged, and this has as a recognition, the glycosylase recognizes it and makes a nick and removes with the help of endonuclease and phosphodiesterase, which is then followed by a polymerase which resynthesizes the lost base, and the ligase stitches it back. So the phenomenological steps will be, again, as you can recognize, the glycosylase recognizes and also acts and gives cue to the endonuclease and phosphodiesterase, which prepares the template for completing the repair, which is then taken off, which is then completed actually by the polymerase, and the final step is ligase. And the ligation happens to be an integral part of all. Because ionizing radiation induces all sorts of lesions, nucleotide excision repair also plays a role, although for the strand break, it is understood that the double strand break repair pathways that I will discuss later has an important role. But because the nucleotide excision repair pathway is somewhat a non-specific repair pathway, which does not get activated following any particular lesions. In other words, NER is activated by any chromatin distortion that takes place due to any sort of lesions in the DNA, and that it gets activated. Therefore, NER can play a role in repairing thymine dimers, predominantly caused by UV, the 6,4 photoproducts caused by UV, say DNA adducts caused by UV, the single strand breaks, the base damage, and so on and so forth. All those lesions which have an ability to distort the DNA. In other words, the conformational changes in and around the lesion sets into motion the nucleotide excision repair pathway. As we will see in this cartoon that I showed, the damage recognition is under the regulation of several gene products, and many of our understanding of the various players in the different repair pathways have come from many genetic diseases with specific defects in particular regulatory component of the DNA repair, has helped us put all these things together and let us know that the DNA repair is actually a multi-gene, multi-step process. And some of these studies have also come from genetic manipulation of cells in vitro. And as I said, much of them are from the diseases or the DNA repair linked disorders, both genetic as well as immunological disorders, that is traced back to defects in the DNA repair. So the strand unwinding is in a transcription factor 2, XPA-B and replication protein A dependent manner. XP, by the way, is the xeroderma pigmentosum, which is one of the UV related skin disease, where the defects are in the different genes whose product plays a role in the DNA repair. For having recognized the nucleotide excision repair pathway works exactly like a tailor to whom you take a piece of cloth, a shirt or a jacket that has got damaged at a particular place, and he recognizes the place where it is damaged and cuts on either sides, removes that, and looks for a similar piece of cloth elsewhere, maybe in the same place or from somewhere else, places it back, stitches it, and gives it back to you, which somewhat looks very similar to before it was damaged. The NER does exactly the same thing using a number of regulators. First, the recognition is by proteins in association with replication protein A, the products of XPA, one of the variants of the xeroderma pigmentosum defects. And then strand unwinding is by multiple members, which are activated up after XPA RPA does the opening up at the site of the lesion. And then that activates the incision by both ERCC and the other variants of XP. Still, the transcription factor 2, XBP, XP are still associated. And then once this incision takes place and it is removed, the resynthesis directed by PCNA with the help of Paul Epsilon and the RFC does the synthesis, and it's ligated by DNA ligases. The excision repair has received quite some attention. There is both global genome as well as transition coupled NER, the global genome NER and the transcription coupled NER, and it is now well established that the TCA, the transcription coupled NER, is a little more efficient and has players which are already an integral part of the transcriptional machinery. The major players, of course, are not grossly different. And what is important in the difference between the global genome NER and the transcription coupled NER are their contribution to the repair of damage in active chromatin as well as not so active chromatin, as also lesions at the site of the nuclear matrix, which is in close association with the scaffold vis-a-vis the DNA away from the nuclear matrix. There are also mismatch repairs, mainly handled by the family of mute, and basic excision repair we have already talked about. As I said, the strand breaks are repaired by more than one type of DNA double strand break repair specific pathways. And amongst them, the major one are the non-homologous enjoining or non-homologous double strand break repair, as well as homologous DNA repair. These have as recognition a cluster of proteins that recognize the lesion, and in a pair 70-80 dependent manner, NHEJ progresses, followed by the preparation and cleaning up of the site by several proteins which open up the DNA. That could be helicases, that could be transcript topoisomerases like proteins, and then prepare the template. And to be able to patch up the newly synthesized strand, the cleaning up of the ends in such a way that there are overhangs are created by other members. And at the end, the ligase 4, preceded by the polymerization, ligates and restores the integrity. In the homologous recombination, and this doesn't need any existing homology of the DNA to be present in order to complete the DNA, to complete the repair. In the homologous recombination, it looks for an existing homology within the genome. Typically, therefore, this kicks in, or this comes into picture in the S phase and in the G2 phase, where the other pair is available and the cell can look for the homology. And similar to the NHEJ, the damage is recognized, of course, by the mRNA complex, and then followed by the RAD protein. And once the homology is recognized, to the broken strand, the homological strand is brought in and inverted, and then stitched after crossover, which is referred to as the holiday crossover, and then the ligation completes the process. Because the non-homologous enjoining do not look for any homology, and also is compelled to clean up the ends on either sides of the strand break, the NHEJ is likely to create a little bit error as compared to the homologous recombination, which looks for a homology and tries to restore the integrity or the constituent of the pre-lesion strand of the DNA as much as possible. And therefore, very grossly, homologous enjoining NHEJ repair is relatively error-prone repair, but it's not that this is always error-prone in nature. And the important players in this are the multi-component, multi-domain proteins that belong to the class of DNA-dependent protein kinase DNA PK, which has both a catalytic subunit and, in case of the NHEJ, the co-heterodimer. There are also alternate NHEJ repair pathways which comes into picture in such of those cells under certain conditions where the co is very minimally functional or available, or have limitations in being recruited at the site. Then there are alternate NHEJs, regional micro-SS single strand elements, and the alternate NHEJ, shown probably not here, is also facilitated by the PARP. It's a PARP dependent or PARP related NHEJ, but again, it is a part of the NHEJ, and as compared to the canonical NHEJ, which is co-dependent, it is generally observed that the alternate NHEJ is a shade more error prone than even the canonical NHEJ. Therefore, if you look at HR, canonical NHEJ, alternate NHEJ, the error proneness increases as we move from HR to the alternative or non-canonical NHEJ. And whenever Q70 and 80 are available in abundance and can be recruited, MHEJ takes the back step. That's why shown our Q70-80 actually inhibits both the alternate NHEJ and the micro-manager NHEJ. There's so much so about the repair of DNA damage. To summarize, many of the lesions, break or non-break type of lesions, are handled by the global, by the nucleotide excision repair pathway, whereas all base damages in an enzyme, the class of enzymes referred to as the glycosylases, which cut the glycosidic bond between the backbone and the nucleotide base, initiated by that, the base excision repair pathway handles the base damage. So base damage, nucleotide excision repair pathway, and double strand break repair pathways consisting of HR, NHEJ, canonical, and alternative NHEJ, as well as MHEJ, are some of the major repair pathways, besides several others. For example, only the reversal, I have not discussed the photolyase, the simple reversal, the trans lesion synthesis, and I did mention only about the mismatch repair and so on and so forth. There are innumerable repair pathways. Perhaps the pathways that are very relevant in case of ionizing radiation-induced damage would be the double strand break repair pathways, as well as to a very large extent NER and the base excision repair pathways. Of course, the mismatch repair is also an important player. Now, radiation also induces interface death, or the radiation-induced damage also stimulates the interface death through apoptosis, which is a P53 and ATM dependent manner. And much of the induction of apoptosis is from basically the damage related activation of ATM and the P53, much less insofar as the execution part of the apoptosis, because apoptosis is also a multi-step process, an initiating signal, an effector signal, and an executor signal, which are taken care of by the regulators. For example, in case of intrinsic pathway, the BCL family regulating the further activator, which is the cytochrome C, And then the effector caspases, which then activate either nucleases or proteases to break the DNA or to reorganize the cytoskeletal architecture by disrupting the protein. That's the execution process. But the apoptosis in this hierarchical development of apoptosis largely is from the generation of signals that activate the regulators. For example, the BAX, it alters the BAX because BAX is a p53 transcriptionally regulated in a p53 dependent manner. But also there are certain transcriptional independent regulation, especially at the membrane, mitochondrial membrane level, which also contributes to the radiation-induced apoptosis. I do not know if I have a cartoon in the next, but probably I should complete here. The membrane damage generating ceramide also is another important contributing pathway for the radiation-induced apoptosis. This is mainly in the cytosol, mainly linked to the MEK and the SAPK and JNK kinases. Again, it's p53 related. The other responses are the senescence and the mitotic catastrophe, which all draw from the activation predominantly from the ATM p53 controlled responses following the DNA damage, primarily through the stabilization from the post-translational modification of these proteins. And insofar as the apoptosis is concerned, it's through the p53 dependent activation of NOx, BAX, and PUMA, which probably should have also, I must mention in the previous slide. But the in a CDK2 cyclin dependent manner, the mitotic catastrophe is activated, and in a RB1 dependent manner, both the involving both the P21 and the E2F, the cessation of cells progressing through the cell cycle will result. While E2F contributes to a very great extent in inducing senescence, the transient cell cycle arrest persisting, which is mainly linked to the P21, could also lead eventually to senescence. The radiation-induced interface death, besides the apoptosis, also has another regulated necrosis, which is called the necroptosis, and that is regulated by a protein called the MLKL, which is actually the mixed lineage kinase-like proteins, which is phosphorylated by a family of receptor interacting protein, RAPK1, shown in this cartoon, RAPK3. In a damage-related manner, the RAPK1 phosphorylates RAPK3, and RAPK3 phosphorylates, in that era, I'm sorry, the MLKL is left out. The MLKL is phosphorylated by RAPK3. Much of the understanding on the necroptosis has come earlier from the studies on the initiation of necroptosis by the interaction of bacterial and viral invasion, including the viral genome interacting with the cell surface receptor. This is by one of the PRRs, the pattern recognition receptors, which then activate the receptor interacting protein K1, and then RIPK3. It appears that following DNA damage, there is induction of necroptosis, which is purportedly by the phosphorylation of RIPK1 and or RIPK3 simultaneously, leading to the activation of RIPK3 phosphorylating MLK. And in a manner that is linked to the ULK family of proteins and a number of ATG members in the autophagy regulating genes, the autophagic response is also induced. Autophagy as a response, as I said, is not necessarily always autophagic cell death. It could also be facilitative autophagy and help the cell survival under certain conditions. Autophagy is seen both as a death promoting and survival promoting response. Oh, no. I'll just cursorily mention about other responses that somebody was asking yesterday about the bystander effect. The bystander effect is I'm sure someone else must have discussed, but if not, the bystander effects, it refers to the response of a cell that was not primarily exposed to radiation. In other words, a response similar to the irradiated cell in a population that was not exposed to radiation. Now, this is very relevant in multicellular organisms, which especially in a solid tissue, but there are more than one ways of interaction between an irradiated cell and unirradiated cell. Now, a number of paradigms have been used to demonstrate the existence of bystander effects and importance and contribution of the bystander effects that includes growing irradiated cells and collecting the secreted products from this in the media, challenging the unirradiated cells with the secretion from the irradiated cells, showing that something that is released from the irradiated cells actually elicit a response which is similar to the radiation response of a cell that is irradiated. Now, this can all be grouped, what is secreted from the cells can all be grouped under one term called the damage-associated molecular proteins, either released as such or put inside or encapsulated inside a vesicle and released into the cells. However, the release of this, all the damage-associated molecular proteins, is also dependent on the on several factors, including the integrity of the irradiated cell membrane. That's where controlled necrosis or the necroptosis also contributes to the bystander effects through a regulated release of DAMP. Of course, besides activating the inflammatory response, which is the activation of the immune cells. In one way, activation by the DAMP of the immune cells is also viewable as a bystander effect. So several studies have shown by co-culturing the irradiated cells with unirradiated cells and observing the pre-labeled unirradiated cells for their response when they are co-cultured with irradiated cells, showing that the bystander effect is indeed a phenomenon. The adaptive doses, the adaptive response is another phenomenon related to the radiation exposure, where a small amount of radiation, very small non-lethal dose of radiation, actually stimulates the cell and stimulates the repair and recover machinery that is demonstrated by its influence on the response of cells which are stimulated by small doses of radiation, challenged with another second higher dose. When you compare the response of cell A, which was not primed with a smaller dose, to that of a cell that was primed with a smaller dose, one observes that the consequences of radiation in terms of any of the endpoints, including cell death, is relatively less in the cells that were primed with small amount of doses. And this understanding was kindled by the observations of phenomenon called the low dose hypersensitive response that has been observed in especially in culture, and to a certain extent also in tissue, where doses much lesser than the 2 gray or 1 gray elicits a higher level of cell death, followed by it is a sensitive response, followed by an increase in survival, and then joining the main dose response curve. Due to paucity of time, I'm unable to discuss these in detail. As I said, there is so much to cover, and I will probably restrict myself to this. The radiation-induced immune response, I guess Dr. Giridhar has probably discussed. I think I will then skip this. So let me now, I don't know how much time did we, it's 11 now, no, 10:50?
Yeah, you have all.
Sir, it is 10:50 only, and you have around 40 minutes.
Another 40 minutes. So let me just quickly go through the cytogenetic damage and the biodosimetry. This is, again, as I said, I want to thank several people, and most importantly, Dr. GIAN, and then material I've taken from awful lot of places. We had this slide yesterday showing that the DNA damage leads to chromosomal breaks, which also shows a dose response similar to the dose response of double-strand breaks. And more than 90% of the breaks are, of course, rejoined, but the remaining leads to consequences. One of them happens to be the lack of repair or a misrepair and an improper restitution that leads to the damage in the chromosome. The DNA double-strand break is therefore the integral component, or it's the DNA double-strand break, which manifests as chromosome breaks in the chromosome or in the chromatid if it is in the G1. In the metaphase, of course, we will see it as a chromosome break. So the misrepair could be only broken ends, maybe in the tail end. It could be in the form of asymmetric and symmetric misrepair. It could be interchromosome and intra and interchromosomal changes, and this is also time and dose dependent. This is the cartoon. You may want to see only the cartoon at the bottom, which is in the white background. The break induced in the DNA double strand when goes through a misrepair and or remains unrepaired and goes through a replication process, you have an aberration. We'll come to several other types of aberration, just one for showing. So when it goes through a replication, then you have aberration here shown as in the form of a dicentric with an associated acentric fragments. So the two small arms are part of the upper arms which they were broken, and these two chromosomes, when they join, you will have a chromosome with the two centromeres, but the parts that were not joined to these two chromosomes will remain as acentric fragments. Therefore, acentric fragments and the dicentrics have a relationship. Now, it's also important the acentric fragments and this because most of the acentric fragments, so much so 70 to 80% of the acentric fragments in the metaphase that can be recognized end up as a chromatin material in the daughter cells following a mitosis, outside the main nucleus, and these are referred as the micronucleus. And therefore, micronucleus is a form of chromosomal, a form of cytogenetic damage linked to the chromosomal aberration, where 70% of the micronuclei is linked to the chromosomal aberration in the metaphase. However, the micronuclei could also be whole chromosome left unincorporated due to mitotic dysfunction and found as a micronucleus in the diaper cells. And therefore, micronucleus is a good cytogenetic damage parameter. Then the dose response curve of the dicentrics follow actually the dose response for the DNA double-strand break, reinforcing the fact that indeed chromosomal breaks are actually nothing but the breaks in the one arm of the chromosome. Now, several things need to be looked into. Therefore, double-strand breaks are the one responsible. So the chromosome is actually a single array of double-stranded DNA. Therefore, any break in one of the DNA double-strand will manifest as chromosomal breaks. If you look at it in the interface, there are methods to look at it. You will see the break in only one arm. When you let this go through S phase and collect them in the metaphase, which is classically done, then you will see the aberrations in both the chromosomal arms. They'll probably skip this. We have paucity of time. There are different types of aberrations. As I said, both the chromosome type of aberrations, where you will see that the aberrations are on the both strands of the chromosomes, both arms of the chromosome, and also the chromatic type of aberrations, where you will see it's only on one of the arms of the chromosome. Now, if you irradiated a cell which is in the G phase, G2 phase, or in the late S phase, especially G2 phase, you will see only this type of aberrations because it has not gone through replication. Whereas if you irradiated a G1 cell and caused a DNA strand break, resulting in chromosome chromatic break, and it passes through S phase, it will generate the chromosome type of aberration. I think we probably will skip this, not going to. So the type of aberrations could be either symmetric translocations or deletions. Symmetric translocations, which was, as we discussed, probably or the movement of the broken pairs is here into this, and then let me just go through. Yeah, okay. The lethal aberrations are the dicentrics, rings, and the anaphase bridges, which hinders the mitosis and as well as progression of the cells through several progenies. So the relationship between other type of aberrations like eccentric fragments, the interstitial deletions, and the cytotoxicity or the cell death vis-a-vis the levels of dicentrics and rings show a strong correlation between dicentric rings and anaphase bridges with the radiation-induced cell death. These are, I think, some more cartoons, which I think I'll skip. Again, the chromosomal aberrations in following a high-LT radiation follows a linear relationship or the exponential relationship, similar to the exponential, simple exponential relationship between the induction of double-strand break and the dose in case of high-fidelity radiations. As we saw with the photon, the double strand break induction is actually linear quadratic in nature, whereas the with the high-fidelity it was linear, and the cell depth is again a shouldered in reciprocal to that, and the cell depth was purely exponential following the high-fidelity. Similar is the case with the linear relationship of the chromosomal aberration or the dicentric induction with high-fidelity radiation as compared to a linear quadratic, which means lower at the lower doses and increasing at the higher dose. The techniques such as the use of probes against different arms of the chromosomes, different parts of the chromosome, and different sections of the chromosome, and tagging them with fluorescent probes has let us gain insight into both the intrastrand crosslinks, the translocations from one to the other, both amongst the chromosomes and within the chromosomes by using appropriate probes. So fluorescence in situ hybridization has contributed a great deal to our understanding of the different types of chromosomal aberrations that ensues following exposure to radiation. And this is also a very powerful technique, unlike the classical banding technique or the light microscopy based techniques with doses as low as 5 centigrade, that's 5 rad here, written as 5 rad, but they're 5 centigrade, that's 1/20th of a grade, revealing the chromosomal aberrations as we can see. All the different colors, the green ones are all the centromere probes, and the other ones with different colors show the aberrations that are formed from different or improper restitution between the chromosomes and within the chromosomes. As I said, the multiple translocations can be visualized using probes against different sections of the different chromosomes, and then using different fluorochromes tagged to this and revealing the different translocations. We'll skip this because I'm not. Now the consequences of these aberrations, as the cells go through different generation cycles, especially in blood, is very relevant. That forms the basis actually for the dosimetry and the choice of dosimeters. So when a bone marrow cell is irradiated, the aberrations that you will see in the peripheral blood, either from the radiation or from the chemicals that are, when go through a symmetrical or asymmetrical restitution, you will see the aberrations of different kinds in the lymphocytes. Now, when a marrow is irradiated, this is all in the nucleated cells or the lymphocytes. When a marrow is irradiated, the damage that is caused in the erythroblast, and therefore a mutation in this, can also cause alterations in the erythrocyte, which has been exploited. That's, I'll come to that later. Besides using the damage to the nucleated cells, whether the peripheral blood cells or in the marrow, that can be queried for the cytogenetic damage. In case of erythrocytes which are non-nucleated, the changes that come about actually come from the changes in the progenitors, and therefore that is linked to the mutation that is caused by radiation, or it is also a consequence of the radiation exposure, which has been considered as a dosimeter. Now, to be able to use any of the radiation-induced changes in the genetic material, be in the form of structural changes like the chromosome aberrations or other alterations in terms of mutations, to be able to consider any of these as a dosimeter, that is to assess the level of exposure of an individual or a population accidentally or intentionally exposed to radiation, it has to satisfy a few criteria. Firstly, it has to be specific to radiation. That, of course, is the most important thing. We should have a good idea about the background of these changes or background of these aberrations, and preferably it should be low. And it should not vary across a population. That becomes extremely difficult. So inter-individual basal variability as well as inter-individual response to this change should be very minimal. And the important thing is to have a dose response calibration so that whenever you have a suspected exposure or a suspected individual exposed to radiation, once you query for the existing chromosomal aberration or a cytogenetic change, a cytogenetic damage in that individual, whether chromosomal aberration or micronuclei or the mutation, the dose response calibration helps you in back calculating the level of exposure, because what you get is the level of aberration or the level of mutation. And if you have a dose response relationship, then you can back calculate and predict the exposure dose. And it should have a persistent effect, and it should be possible to query this without too much of an involvement. It should be easy sampling, ideally body fluid. And the faster the result, the better it is. Less costly, of course, it's more preferable. It's used in triaging, in the sense, if you have a population suspected to be exposed to radiation, one would like to know, based on either the information or a quick overall assessment of the dose in that area. You may segregate several hundreds or thousands people purported to have been exposed to radiation, but you cannot probably meaningfully and gainfully use all the resources and help those individuals who actually need attention, who actually need intervention, who actually need medical management. So biodosimetry helps in confirming the individuals exposed, as also the possible levels of exposure vis-a-vis individuals who are not exposed or who are exposed way below a level that requires attention, whether it is segregation, observation, intervention, and hospitalization, which is called the triaging. To be able to make this decision amongst a cohort who are supposed to have got exposed or purported to have got exposed, biodosimetry helps in triaging, segregating, so that people who actually need attention receive attention and do not consume resources in cohorts who did not need and deprive the ones who needed. That's why biodosimetry is a very is an extremely important component of the medical management of radiation exposure. And the last point that I put in here is probably what is important, because biodosimetry comes into picture when there could be exposure to large number of subjects. If it's a large area, a city, or at several places when there is an exposure that has taken place. For example, what happened in Chernobyl, an accident affecting a large number of people, or unfortunate, God forbid, that doesn't happen. Whenever there is a nuclear detonation, large number of people would be potentially exposed to radiation. the possibilities of handling all the samples for doing biodosimetry at one place is unthinkable, it is impossible. So there will be a great need to handle the large number of samples for analysis at several places. It could be within the country, within a state, within a city, but across the globe. Therefore, it is extremely important that we have a harmonization across different labs, be it different labs in a city or across the globe. There should be harmonization. What do I mean by harmonization? The way the assay is done and reported should be same across all these places. The reason being, whether the sample is done at lab A in country A, or lab B in country B, or lab C in country B, or so on and so forth, the results should be same. So that when there is a need to handle large number of samples, it should not matter where it will be done. Therefore, harmonization is an important aspect of dosimetry in the medical management whenever we face with exposure of large individuals. So there is a great deal of interest in networking, not interest, there is already existing networking. The European Union has a network. They have identified several labs which will do, and periodically these labs keep doing the exercise. Sample from one lab is sent to the other. Preparation at one level after having collected is sent to the other. And slides from one is sent to the other whenever the dicentrics are stored. So all harmonization at various levels of the assay, sample collection, processing, culturing, harvesting, staining, analysis, all these things are paid attention, and the exchanges are made at various levels from different labs to ensure no matter where it is done and which part of this whole process is done at different places, it is reliable. And as of now, the gold standard that is used for biodosimetry is the dicentric chromosome analysis, or the assay for dicentric chromosome, because it is truly a radiation-induced chromosomal aberration. Of course, whenever we have a suspected exposure, a micronuclei is equally good. Whenever we know that some individual is exposed and we want to confirm, other assays are also used, like, as I said, the mutation and so on and so forth. But as a gold standard, the radiation-induced dicentrics is very extensively used. Because of the need for handling large number of samples under these extraneous conditions, a lot of attention has been paid in establishing automated methods for handling large number of samples. These all have automated liquid handling, processing in a high throughput manner. So at several places, including in New York and also in Bethesda and several other places, robotic based and also highly automated sample and liquid handling systems are in position, which can handle samples to the tune of many hundreds to many thousands in a day. Now, depending upon the time and the need and the level of exposure, we can think of different biodosimeters. So one can actually look at the changes in the blood cell count, because the blood cell count keeps going as a function of time. I'll come to this a little later. Is only to suggest that the simplest way, of course, is to just get a white blood cell count and get a rough idea of whether an exposure has taken place or not taken place. It's possible. In this I think I'll skip this. This I don't need it here. I'm sorry. Okay, we got it. So the best possibilities that we have for the biodosimetry are to look at the cytogenetic indicators. As I said, most reliably and truly are the dicentrics and micronuclei. Translocations are also in some respect, because I have a cartoon which will give an idea about where to use when. And it is also possible to get the radiation-induced cytogenetic damage, not having to culture and just do the cytogenetic damage in the pre-mitotic cells, and it is a technique called the premature condensation or the prematurely condensed chromosomes, which I'll come to. I think we will not need this for want of time. Now we will skip this. This we have already discussed. We will discuss. Okay. Now there are. This is a cartoon that I've taken, which gives us different alternatives, which is summarized in a short form in the next one. So the damage induction, the cytogenetic damage, as I said, relates back to the DNA strand break, which can be immediately queried using strand break assay, typically gamma HTX assay, because of its strong relationship with the double strand break and ease of doing, and ability to automate using flow cytometry or image analysis based. That's being strongly considered, has its own merits and limitations. One can do an exposure related changes in the genome by looking at the gene expression changes, which can be done immediately. And if the cells are allowed to then go through a cycle, then one can do the classical cytogenetic damages in the form of dicentrics, rings, and acentric fragments. And in the pre-mitotic or in the metaphase, just before the division, or as I said, can be queried as a micronuclei. And this is how it will all appear. And this is an overview that I just put together. The DNA damage H2X loci can be done immediately. We don't have to wait. If somebody is purported to be exposed or suspected to be exposed, immediately you can take the blood and the damage can be assessed using the H2X loci. Gene expression can be done immediately. The premature chromosome condensation, I don't know if I have a cartoon, maybe not, then I'll finish the discussion here. What we do here is we do not have to grow the cells in culture and let them be harvested after adding a mitosis arrestor or compounds like the cytochalasin B, by using the colchicine. We don't have to do that. We don't have, typically it takes 48 to 72 hours for the cells to come to the metaphase. You collect them by adding colchicine if you're looking at the metaphase. Or if you let them divide, then you add a cytochalasin B and collect the binucleated cells. You let the nucleus divide, as you will see in the bottom cartoon, but you will not let the cells divide and score aberrations, score micronuclei, which is a consequence aberration, in the daughter cells. But both of these, the classical one, the dicentric assay, the bottom, and that I am showing here, where is it? Yeah, and here will require 2 to 3 days before you can do the assay. However, PCC can also be done quickly, even after irradiation, in the interface, in G1 phase and in the S phase. Typically how it is done is using factors that are found in the metaphase. You provide that to the interface, and this is done by fusing a cell which has a high rate of mitosis, like the Chinese hamster ovary cell is typically used for this. You make a fusion of the lymphocytes soon after irradiation with the CHO cells. Here, these are the CHO cells. And the factors that were contained in this, when these were fused with these cells using certain compounds like the polyethylene glycol, which fuses, the mitotic promoting factors from the CHO cells are provided to this. The chromatid starts condensing, somewhat comparable to the chromosome at the time of condensation in the metaphase. And then it lets you look at the fragments you can see here. If you did this for unirradiated cell and irradiated cells, you can score the number of fragments. What PCC lets you do is look at the increase in the number of fragments, the basal fragments, the basal number of chromatids, and as a function of those, the fragments number will increase. Then you can generate a response between dose and the increase in the fragment size after fusing. And these assays, as I said, can be done in hours, so an hour after irradiation or even up to days, it is viable. Now, dicentrics are very good. Oh, I'm sorry, I made a mistake in pasting. Dicentrics are good, and micronuclei is good from up to about several months, because the cells with these type of aberrations will be eventually eliminated. You can't do a dicentric assay after a year or two after exposure. But assays like the translocation, which are stable aberrations, can be employed. Both these are wrong, please. Days and weeks are not correct. This is immediate, this is few hours, and this is up to several months, and these are also up to several months, from few weeks to several months. You can also do, besides the cytogenetic damage-based assays, you can do the radiation-induced mutation at different loci. One is the glycophorin A loci, I'll come to that, and T-cell receptor mutations and HPRT mutations. I think we'll skip this, we don't have really time for it. So I'll just touch upon the glycophorin A, TCR, maybe HPRT I don't have. The glycophorin A mutation is actually is an assay where radiation-induced loss of allele is acquired. Glycophorin A is actually a RBC cell membrane protein, siloglyco. It's a glycoprotein containing sialic acid, and it actually plays a role in the grouping of blood grouping through the M and N. This is an autosomal locus found on chromosome 4, therefore it's a 4L and then M, and with 2 alleles, and these are designated as M and N, and the product of this gene is, as I said, is on the RBC membrane. Because of the mutation that occurs in the progenitor cells of the RBC precursor, the progenitor cell, the mutation manifests in loss of allele in the RBC after the terminal differentiation into RBC. And the glycophorin A is also called as now the CD235A, which because it's a cluster of differentiation protein. The frequency of these variants increases with exposure dose, and mainly, as I said, due to the mutations in the progenitor cells. It can be easily identified using antibodies against this and quantified, and it can be rapid by using the flow cytometric assay. It can therefore be quick. And most importantly, it can be retrospective. A glycophorin A mutation can be queried anytime after exposure has taken place. This summarizes a little more information. The sensitivity threshold, of course, below 0.2 gray, it may be difficult. It merges with the basal levels of the variant. And this is only possible in individuals heterozygous to this loci. That means it's only in 1/2 of the population this assay can be considered. So in such of those individuals who are heterozygous to this, an exposure can be quickly identified by looking at the possible mutation in the GPAs. I guess this is too crowded. I'm going to leave this. It's only to show the mutations in the T cell receptor is also a dosimeter that has been used, although not widely. Shown here is the data for a highly exposed, weakly exposed individual and a control cohort, not individual, control cohort. You will see that the weak exposure doesn't really discriminate much from the control, but a highly exposed level does show a significant increase in the rate of mutation. And patients who receive radiotherapy for various anatomical locations, interestingly also show that the HPRT location mutation suggesting or confirming. In fact, this confirms, because if you look at only these two, one would wonder whether it is really useful. But in patients undergoing radiotherapy, we know for sure that they were exposed to radiation. And typically partial body radiation, because they would receive a different radiation at different parts of the body depending on the anatomical locations. You know for sure that they were exposed, and you do a TCR mutation assay, good enough, you will see this. And when now you see mutations in suspected individuals, a higher level, you know that indeed this patient, in these individuals, there was exposure that had taken place. I think the right one is the HPRT location, HPRT loss emitter. I think we will probably not very much. I only wanted to draw your attention to this example from work that was published some time back, where they queried in two mouse strains, the black mice and the Balb C mice, both the T cell receptor mutations in the T cell, and simultaneously micronuclear frequency. You will find that at least under these conditions that they did at 2.5 gray, your micronuclei is as good, if not better than the T cell loci mutation. So one probably need to consider several aspects before employing a particular approach for the dosimetry. I guess I will probably stop here. I probably at 11:25, right?
It is 25.
11:25. So I think I'll stop here. I'm sorry for rushing through, but I had to, and I'll be willing to make an attempt to answer questions.
Thank you, Dr. Dwarkanath. A wonderful talk as usual. I think instead of reading from the chat box, I think I will request the participant to ask a question directly. I think that is better for the interactions and communication. Yeah.
Alec, I think you can ask the question.
Good morning, sir. I wanted to know what is, you talked about low donor variability and the persistent effect.
You say that again?
Donor variability and the persistent effect.
Yeah. What do I mean by that?
Yes, sir. I couldn't understand that.
Okay. For example, this is practically very important. Suppose the dosimeter that I am going to employ, and let's say I am going to employ, for example, a particular type of mutation, okay, or a particular type of chromosomal aberration as a measure of finding out whether these individuals have got exposed, and if yes, what is the likely level of exposure of these individuals. Now, for me to be able to report this reliably, using a biodosimeter, the following amongst other things are important. As I said, inter-individual variation and persistence. That means if I find that the type of aberrations that I am examining, even in a population, the basal levels are so variable. Because you take the individual, you will only query him for the first time. But if the parameter that you are using amongst the unexposed individuals have a huge variation, and the induced changes are probably very minimal, so you wouldn't be able to confidently say this individual is exposed to this level because of the large variations in the basal level of the parameter that you have used for assessing the exposure. That's the inter-individual variability. Second one is the persistence. Now, if you choose a dosimeter, and this dosimeter is chosen, and you have chosen a particular dosimeter at a particular time that you want to investigate it after an exposure. Now, if it persists up to the time that it is valid, then the chances of success is very high. I also showed where there were some errors because I made probably an error in cutting and pasting. I put a time limit where each of these dosimeters are useful or relevant or applicable. Now, as long as you use in that limit, it's reliable. But if you use these aberration, if these parameters beyond the time that you can query them or much before that they actually manifest, then also you will infer incorrectly. The second aspect of the persistence is that this change is seen in n number of individuals without any exception every time you query. And these are typically done by irradiating several animals, because insofar as these responses are concerned, it is human or animal, this is one and the same. If you find that persistently, whenever you expose a group of animals to this dose and query at that particular time, you get the same level of the damage there, then you have confidence that this damage is consistently and persistently found under these conditions. And therefore, this could be used for assessing the likely exposure. But when is biodosimetry used? It is used in a scenario where somebody is supposed to have got exposed, suspected to have got exposed. Number one. Number two, suspect and exposed to what level, which determines what kind of attention he will get, what kind of medical management, and how quickly he or she needs the medical management. And for that, the exposure certainty and the level of exposure identification becomes very important. And this will be must if you choose a parameter which has a large basal value variation and is not persistent, that one time you get, the other time you don't get. That's all what these two means.
Okay, thank you, sir.
So if there are no other questions, I would take this opportunity to thank Professor Dwarkanath, who has really given immense knowledge in this 1.5 hour session, as always. Thank you, sir.
Pleasure. And I'm sorry for cramping up the things.
No, certainly, this was one of the most educative sessions. So now, with permission of the organizers, shall we move to the next section of today's sessions? So
Yes, sure.
Okay,
Sure.
so the next session is a tutorial which will be handled by Dr. Anant Narayan Bhatt and Professor Vibha Tandon. So let me introduce the session and the faculty for this session. The session would be on the demonstration of tissue section showing radiation-induced damage in different organs and their protection, and also the monitoring and damage assessment. So I'm sure this tutorial is going to be of immense benefit to all the students as well as the established researchers. So I'll now like to introduce Dr. Anant Narayan Bhatt, who's a senior scientist at INMAS, New Delhi. His research interests include the cancer cell radiation biology, and he has an illustrious career starting with a PhD from Central Drug Research Institute, Lucknow. He worked as a postdoc EMBO fellow at University of Zurich, also worked as a Fulbright fellow at Johns Hopkins University, United States, and currently is also a visiting faculty at the Department of Zoology, University of Delhi. And recently, he's been awarded with the Laboratory Group Technology Award and the Laboratory Scientist of the Year Award in 2022. Dr. Anant has authored several international scientific publications, and some of them have received tremendous number of citations as well. So his research papers have been quite impactful. And last but not the least, I would like to tell you all that he's been a mentee of Professor Dwarkanath, another legacy of Dwarkanath sir in various institutes across India. And the second faculty for this session is Professor Vibha Tandon. Professor Vibha Tandon needs no introduction. She's the organizer.
Yeah.
And everybody knows her. She's very well established in her field and has contributed immensely, not only to the research radiation biology field, research foray in the radiation biology field, but also in creating a legacy of students and trained faculty members in various institutes of India. So over to you, Professor Vibha Tandon and Dr. Nath.
Yeah, sir, you have to stop sharing.
Yeah, I was going to say, may I not only stop sharing, but also leave for the time being?
Yeah, sure.
I have a commitment, I'll come back.
Sure.
Thank you.
Yeah. So Dr. Anant is here, I think. Anant?
Yes, sir, you have, I think still it is shared. Dr. Anant, sir.
Oh, it is still shared?
Your screen, yeah.
Wait a minute, sorry. 1 second.
Yeah,
sorry. Yeah.
So Anant will start the show.
Okay. I'll beg your pardon.
Yeah.
The slides are shared. I mean, can everybody see the slides?
Yeah, we can see.
Yeah. Okay. Thank you.
Anant, you have to go to the full screen mode.
Is it there, ma'am? In full screen mode?
No, it's not.
No, it.
Actually, there is some maybe bandwidth problem.
Otherwise, go ahead because you can read the words.
Okay.
Yeah, there's no problem.
Okay, so I will go then probably close this so that
now it's okay.
Okay.
You can increase the size. Yeah. Yeah, little bit less. Yeah.
So thank you, Dr. Ritu Gupta, for a nice introduction. As we discussed yesterday about the cellular effects of radiation. So now moving on to when the cell gets damaged, it is part of some tissue, and then it will lead to, as I said, that whatever cellular damage will happen, it will pour lot of oxidative molecule. I mean, because of the radiation is induced oxidative stress, lot of macromolecule will get oxidized, and they will actually released once the cell will die. So they will release to the neighboring cells, and neighboring cells will slowly having this effect, bystander effect, and then they will, this will lead to inflammation in the neighboring cells, and then the neighboring cells will die. So what are this also I showed yesterday, what are the different mode of exposure? I mean, people can get exposed from a distance, or if the radioactive material itself got ingested in the body through I mean food material or through inhalation or whatever. I mean there are different ways through which it can go into the body, and then it deposits there and continuously giving I mean irradiating the neighboring cells. So because of that, the damage will happen and it may lead to different places. So basically, this is a slide. What it is showing, if you see a small picture here on the left side, this one cell is dying and then it is releasing. This is I mean a blood picture taken from a blood smear, and you can see this cell is dying on the right side, and it is releasing a lot of things which may actually cause lead to inflammation basically. And once the tissue will damage, it will pour a lot of things to the blood, which will take the I mean these inflammatory materials to different tissues which are not yet got exposed. So to radiation, in case of focal radiation, in case of cancer treatment, and when the person got exposed also to different I mean different tissue, not I mean irradiated focally, not irradiated by accident, also not the whole body irradiation he has received. So in that case also the damage will happen in one part of the body, and it will travel, it may go to the other part of the body through circulation and inflammatory materials. So those inflammatory materials will go and cause death at different level. So today we are going to talk about the damage which is happening to the tissue, and the molecular level damages. I mean, the students have gone through, I think, extensively now. We have covered yesterday molecular level damage. So what are those molecular level damage, and how you can counter? Once you will counter that molecular level damage at cellular level, then the cell death will not occur. And once cell death will not occur, the inflammation will stop. So how it is reflected at systemic level is inflammation, leukopenia, and then sepsis is one thing. Like, as I said, if gut damage will happen, microbes will enter into the bloodstream, then sepsis will happen, and that leads to the mortality. So if you counter at cellular level by modifying the signaling, by protecting the macromolecular damage, radiation-induced macromolecular damage, then cell will be protected, and then the entire things can be protected. So coming back to what basically happens, I mean, when the radiation, people get exposed to radiation, what basically happens? So here, when we are studying the tissue damage mainly, then we have to understand in different form. Like if the exposure is very less, I think I could not attend the entire thing. Probably the stochastic and deterministic effect of radiation would have also been taught. So when the radiation dose is minimal, so the chances is very high that the person will have the stochastic effect. So stochastic effect will lead to basically, I mean, the thing which you cannot determine, I mean, the cancer and other things, if at a very early low doses or even at high doses. But the deterministic effect, which we can see here, that if person has received some amount of radiation, let's say 1 gray or something, then we can say that the leukopenia will occur, so that you can determine the dose and causal relationship. So at very low doses, person will show late effects of radiation, which we will be talking about. Some of the late effects of radiation at higher doses, also lung damage, but a lot of late effects appears when the person receives the radiation at low doses. Then slowly, when the radiation dose will go high, you can see on the x-axis, then what will happen? Hematopoietic syndrome will occur, GI syndrome will occur, and these symptoms will appear in within few days only. I mean, within week time only, person can see if the radiation dose goes high. And further, if radiation dose goes high, then CNS disturbance will happen, and this kind of people die generally within few days. I mean, maybe in hours or within few days. And if the radiation dose is very high, very, very high, then molecular death happens, and that happens within few hours. So this graph basically suggests that how with different doses of radiation, the different kind of tissue, the susceptibility of different tissues is different in the human body, and they start getting damaged, and they show the response, differential response to radiation. So this also we have discussed yesterday. So predominantly at low doses of radiation, I mean, when we are talking about anything less than 9 gray, 9-10 gray in case of mice, and 5-6 gray in case of humans. So you can assume that predominantly what basically happens, that hematopoietic injury happens, which happens even after 1 gray itself. 0.7 gray onwards in humans, hematopoietic injury has been observed. And then hematopoietic injury will happen that will deplete all the cells, I mean white blood cells predominantly, because RBCs are relatively more resistant, but RBC lysis also happens if the dose is high. Then GI disturbance will happen at little higher doses, maybe 3 gray or above. The person 3 or 4 gray above in humans, and in mice and all, the classical dose is 9 or 10 gray to see the GI damage, focal irradiation, or whole body irradiation. Also, you can start seeing anything above 7.5 gray, 8 gray GI damage can be observed. So these cells are why they are relatively more susceptible. Why? Because they are proliferating relatively faster, because blood cells are continuously dying. RBC has a life of 120 days. WBCs also have their own life, so they will die, and then in bone marrow, the formation of new cell will happen, and they will come and replace. So these cells are continuously proliferating, and because these cells are continuously proliferating, similarly as cancer cells, these cells are more susceptible to radiation, and that is why radiation is used in cancer treatment also as a radiotherapy, because those cells are also relatively fast proliferating. So the bone marrow will, the depletion will happen. Leukopenia can be observed if the person got exposed to 1 gray or 2 gray radiation. Next day, if you take the blood count will be significantly low. That is called nadir. Very minimal WBCs can be observed in the blood. Similarly, at relatively higher doses, these GI cells, crypt cells are also very sensitive, because they are also continuously proliferating, and because of that, the GI damage may also occur. And then after some days, what will happen basically, the person will have diarrhea. The symptom of diarrhea will come, and that can be seen as one of the symptom of radiation-induced GI damage. So this is a image which is taken from a nuclear plant. This person was working in a nuclear plant. You can see the top image is from the Hiroshima Nagasaki blast, I believe, and you can see that the burn, skin burn can be seen very clearly. That is radiation-induced skin burn. In the lower images, what happened, that this person was working in Iran in a nuclear plant, and this fellow has taken a iridium 192 rod, and he took it, he kept it in the pocket, and then he probably trying to steal or whatever. So in the first image, which is taken I think 1 day or 2 days after, you can see that there is hardly anything can be observed. Little later, if you'll see, this is day 11, you can see the middle image, that is on day 11, and erythema can be seen. The skin became completely red. On the right side, you can see the burn on the skin. So this is basically, this is called cutaneous radiation syndrome. And if the radiation has been given from outside, this burn can be seen. And this is a very common thing you will be seeing. The patients who undergo radiotherapy, head and neck cancer patients undergo radiotherapy. Generally, very high doses of radiation in different fractions are being given, and you can see that they show that complete area becomes black. So it is not up to this level. Cutaneous radiations burn cannot be seen up to this level, which is seen in this patient, this person. But radiotherapy patients mainly head and neck cancer patients, they also show blacklessness of the skin in and around area. So this was a cutaneous radiation syndrome. Now we'll be discussing mainly about the acute radiation syndrome. So acute radiation syndrome, as I said, like once the person got radiation exposure anywhere between 1 gray to 5 gray, 6 gray, like that, for a human, and for a animal or mice, I'm talking about C57 mice, which we use in our lab routinely. That for that LD100 dose is somewhat like anywhere between 8.5 to 9 gray. So so up to, I mean, GI damage, you can see 7.5 or 8 gray onwards, and hematopoietic syndrome, you can see 2 gray onwards. Generally, we give 2 gray radiation to observe any hematopoietic syndrome. And as I said, like if the exposure is very high, then CNS disturbance will happen, and then the person may die within just few days. So, but these animals, they generally survive up to 15 days, 20 days. If you give a 7.5 gray radiation or 9 gray radiation, C57 animals, they survive for 15, 20 days. Not, I mean, if we have given LD100 dose, then generally they survive up to 20 days, and then they anywhere between, they start dying from 12 days, and the last death may happen on 20 days. Of the complete, I mean, let's say if 10 mice were irradiated, so all the 10 mice will die within 20 to 25 days completely. So I mean, hematopoietic syndrome can be observed in this range of time. And then GI syndrome also. GI syndrome generally happens on the 3rd day onwards, and we will be seeing that when we will be discussing individually about these syndromes. So hematopoietic syndrome in humans, it generally occurs anywhere between 0.7 gray onwards. And you can see here on the left side, these are the different cells which are susceptible to radiation. And on the right side, you can see in the sham, there are a lot of proliferating cells. This is the section of the bone marrow. And you can see that a lot of proliferating cells are there, which are blue color nucleus stained. So they are proliferating cells. But in a 9.5 gray exposed animal, you can see that the bone marrow section hardly has any cell. So majority of the cells are dead. So the stem cells are itself. I mean, if you monitor the blood count, the blood count is completely gone after 1 day, 2 day. And this is a 1 week after. If you take even the bone marrow section also, the bone marrow cells are also completely gone. So the stem cells, there is hardly any stem cell to produce at such a high dose to produce the proliferating cells which will come to the blood. So the white blood cell will not come to the cell to the blood, and because of that, this leukopenia happens. And if it persists for long, if cells are not able to, if very minimal stem cells are remaining, then they will not be able to produce these proliferating cells, and then the person will die because of this kind of person is like, they don't have any immune system because white blood cells are not coming to the blood. So immune system is not there, and the person may die because of that. Opportunistic infections. So this is a study from the lab. Here you can see that we have exposed to the animals 7.5 gray radiation, and you can see here that bone marrow cells which we have count. You can see that in radiation, which is the middle bar, you can see that the bone marrow cells are significantly low as compared to control. But when this is treated 2 hours before the radiation was given, you can see that the recovery is a significant increase in the recovery of the bone marrow cells can be seen. And this is a bone marrow section after 1 week. On the left side, you can see it is full of blue color, which is indicative of proliferating cells. And in the middle one, you can see that the blue color is, there is hardly any blue color. So, the stem cells are not there mainly to produce WBCs. But on the right side, which is treated with drug, you can see relatively more proliferating stem cells are there, which helps in replenishing the circulating blood cells, generally anywhere between 14 to 21 days. it comes back. I mean, the level of the cells comes back in 14 to 21 days. This is another study with another molecule. Here you can see that on the left side that this drug was given, and when the drug was given 2 hours prior intraperitoneally, and then the mice were exposed to radiation, and you can see here that there is significant increase in the decrease in the death mortality in the mice which are treated with radiation. So the green line shows the mice which are treated with radiation, treated with drug and then given radiation. And on the right side, B, you can see the total WBC count. There is a recovery. I mean, the fall itself is not that much, because red line, you see that it has almost gone. WBC is like 1 into 10 to the power 6, this value. And then this is almost gone. But slowly recovery happens here. But in the green line, if you see in the graph B, the recovery is happening. I mean, here also the death happened significantly, the damage happened significantly, but slowly the recovery is happening, and then the cells got recovered completely. And these are individual lymphocytes. You can see lymphocytes are very susceptible to radiation. So lymphocytes are also recovering, then granulocytes are also recovering. Platelets and, as I said, platelets and RBCs are not that susceptible to radiation. So not much decrease has been observed in platelet and RBCs. You can see here, but there is relatively better recovery can be observed in this also. So now coming back to GI syndrome, that is gastrointestinal damage which happens. So if you give little higher dose, I mean, to the animal or human got exposed to relatively higher dose, then what happens, because as we discussed yesterday, ROS induced injury will happen, then enterocyte depletion will happen. The cells will die because of enterocyte cells will die, and that depletion will happen. Then mucosal barrier breakdown will happen, and then mucocytosis will happen. This will lead to the diarrhea. And like gut is full of microbes. So suppose that if somebody, some person got 4 gray or 5 gray exposure, or some animal got some 7-8 gray exposure, what will happen that the immune system is already gone, I mean, because the WBCs are gone completely at this dose. And GI is also getting damaged. So all the microbial microbial population which is there, which is good if it is there in the gut, but it should not enter into the blood. So this damage will happen, leaky gut will happen, and that leaky gut will allow the microbes to enter into the bloodstream, and that will lead to adverse tissue remodeling, and sepsis also will happen, because immune system is not there to protect it. So to fight with those microbes. So sepsis may happen and the person may die because of this. So here you can see, as we showed, that the crypt cells are, so this is intestinal crypt. This is studied in duodenum. So you can see, as compared to control, the 9 gray 9 gray exposed mice, you can see the basements are gone completely, and these are very small villi. You can see the villi become very shorter, and a lot of damage happens to the villi. However, the mice which are treated with drug and then given radiation, you can see the relatively longer villi can be seen. And in the lower panel, if you'll see, this is a BRDO staining, and you can see here the mice were given BRDO before they were sacrificed for this analysis. You can see that in the middle row, the IR9 gray doesn't show much green color. However, in the drug treated mice, it shows very good green color. So which is suggestive of that the proliferation is happening. The crypt, proliferation is happening in this this gut section, and which is suggestive of that the drug is able to recover the radiation-induced damage to the GI cells. Now, these, as I said, like these things you will observe like within few days, like within week. Generally, if the radiation dose is not very high, the GI damage takes hardly 7 days to 10 days to recover. So GI will regenerate, and then the damage, the mice may recover. If it is not recovering, then that mice will die. These things generally occur within 10 days or within 15 days maximum. And then you would have seen like the 7.5 gray exposed, the radiation, blood cells hematopoietic recovery is also, even if you have not given any drug, slowly it starts recovering. And if the mice or subject hasn't got any opportunistic infection during that time, then the chances of survival will be there if the radiation dose is not very high. However, what happens, like some of the organs like lung is also relatively in the next line. It is susceptibility in the after blood, hematopoietic and GI. The next susceptible organ is lung, which is relatively more susceptible to inflammation. So what happens, like generally it is observed in mice that if the person got exposed to radiation, and mainly like localized in the thoracic region, then what will happen in 3 months, radiation-induced pneumonitis will happen. And that pneumonitis, because it is a chronic inflammation. It is not a, acute inflammation. So since it is a chronic inflammation, what will happen? It will induce fibrosis. So generally, after exposure, at 3 months, people develop radiation-induced pneumonitis, and at 6 months, people develop radiation-induced fibrosis. So these are two classical examples, of radiation-induced lung injury people have observed. And you can see on the left side, the lung is on the left top, the lung is completely healthy. However, slowly you can see that it is becoming a rotten kind of thing. The lung become completely fibrosed. So these fibrotic lungs, because a lot of collagen deposition happens in these lungs, and so the area which will become fibrotic, that becomes useless. The respiration doesn't take place in that area. So the efficiency of the lung will continuously go down. And suppose that if 50% of the lung of somebody has got fibrotic, then the person is basically respiring with the 50% of lung only, and that too inflammation will be there continuously, and that will also not be, slowly the capacity of the lung for respiration will go down. Beside this, the long-term effects are seen as radiation-induced thyroid cancer and blood cancer, especially if iodine, radioactive iodine I-131 is there. Then what will happen? It will be ingested and it will go and deposit in the thyroid glands only, and then there it may cause thyroid cancer, and sometimes these exposures leads to blood cancer also. So while talking about radiation-induced lung damage, you can see here this paper got recently accepted. On the top row, you can see that A is basically a CT image of the mice, where you can see like the control is perfectly all right, and the drug is also perfectly fine. But in the radiation exposed, the third column, you can see here radiation exposed, it has a lot of patches of fibrosis, which is like when it was pre-treated with the drug, it has gone. This drug was also given intraperitoneally, and this fibrosis is gone. This image was taken on 6 months after exposure to radiation. And this is the lung morphology. You can see in the third column in the B lane, if you see that there are small white patches. These patches are basically fibrotic areas of the lungs, radiation-induced fibrosis, and which cannot be seen here in the right side. On the last column, which are treated with drug before irradiation. This is in C lane, you will see the H&E staining, and this slide, similar slides were stained for this is called Picoserious stain, which stains basically the collagen. And you can see that there is a lot of collagen deposition happened, and these collagen are basically responsible for fibrosis. So the lot of collagen deposition you can see here, which is not there in the drug plus radiation group. So that means if you give any drug which is able to inhibit this damage first thing, and then can modulate the inflammation at 3 months, then what will happen? This chronic inflammation will go down, and that going down of the chronic inflammation will not lead to the collagen deposition and fibrosis at 6 months. So this fibrosis is reversible. These are certain other markers also. On the right side, you can see the bar diagram, which is showing the hydroxyproline. Actually, hydroxyproline is one of the amino acid which is very prominent in collagen only. So you can quantitatively also measure in the lung that per gram how much hydroxyproline is there. That is a measure of collagen indirectly. So this data also corroborates with that. Here we have again, this is a work which we have done at Hopkins. So you can see here that after radiation induced, we wanted to see that what are the different kind of damage associated molecular patterns, and we found that oxidized phospholipid is one of them. So you can see here all the, in the top IHC stain at 14 weeks. This is 10 weeks after the radiation exposure, and after 14 weeks you will see small brown color spots are there, which is suggestive of oxidized phospholipids are started being produced there from dying cells basically. And after 23 weeks, you can see here that lot of patches of oxidized phospholipid can be seen on the top, and the lower is again Picoserious stain, which is again suggestive of more and more what is called the collagen deposition, fibrotic lungs. Then we measured the oxidized phospholipid with quantitatively also, and we have shown that at 15 gray the oxidized phospholipid level in the lungs was extremely high, and probably this is one of the driver of the chronic inflammation as well as the fibrosis in the lungs. So these are the markers which can be studied, that how it is happening. Then in our lab, you can see here on the left panel completely that when you expose the mice to radiation, we use generally C57 mice, and you can see here that the graying of the hair can be seen here in the mice. However, when it is the mice were given the drug DNP, then you can see that the graying is relatively less, and this is another protector molecule which we have identified in our lab. And you can see here that basically what happens, radiation damages to the keratinocyte stem cells, and that is basically responsible for discoloration of the hair, and these drugs are able to protect those keratinocytes. So similarly, like in GI, when the stem cells are damaged, then the crypt will be there will be a lot of damage to the crypt. So similarly, if you protect these stem cells also, then probably the discoloration of hair will not be that will not be that prominent in those mice. So relatively, you can see the whole body hair discoloration is seen here. However, in these mice which are treated with DNP, only in the area of head only you can see the discoloration. However, the remaining part of the body, you won't see much discoloration of the hair. So what are different kind of drugs which generally, like in our lab also, we are also working, and what are different kind of drugs which can be developed in case of this radiation exposure. So radioprotector is a drug which is given before, like this can be given only in case of planned exposure. Like I know that after 2 hours I have to enter in an area where there is a radiation. So I take this drug, and then enter to the area of radiation where the radiation contamination is there or radiation exposure is there continuously. So that those drugs are called radioprotected drugs. And then if you don't know, I mean, in case of unplanned exposure, suppose that I already got exposed to the radiation, and tomorrow or after some time I realize that that I got exposed to radiation, and then I take some drug, that those drugs are generally called radiation mitigators. So, you know, like these drugs have been taken before symptoms appearance. I mean, we just know that we got exposed to the radiation, but there is no symptom. So that is we have taken as a safety measure. So these drugs are called radiation mitigators. And these drugs, I mean, there are a lot of groups which are involved in developing this kind of drugs also. Then if the symptoms are already appears, either in case of planned exposure or unplanned exposure, if the symptom occurs and then you go for the drug, those drugs are called therapy basically. So there are three different class of drugs which are there in these, in case of handling the tissue damage, radiation-induced tissue damage. They can be radioprotected, radiomitigator, and therapeutics also for protection from radiation-induced damage. So this is like in our lab, we screen and then find the lead molecules through different assays, and then we basically develop. You can see here we have developed a lot of, I mean, these are the assays you would have already seen in different presentations, that if the cells are untreated, you can see a lot of colonies are being formed. However, in case of radiation, you can see the number of colonies formed is very less in the second column. In the third column, this is another drug. When given the radiation, you can see the number of colonies formed is relatively high. So that means the protection is there. And in the third one, again, there is another drug which again shows that the protection is there. So then you plot and then you calculate the surviving fraction. I think Dwarkanath sir would have already talked you all about this in detail. And you can see here that the red line is drug treated. So surviving fraction has increased, which is suggestive of that the drug is protecting against the radiation. This is what we do in case of animals, like we treat the animals. We are generally involved in development of radioprotectors predominantly. So we give the drug treatment before, but we are working on the mitigators also. So in case of mitigators, we generally give radiation after exposure, radiation exposure, 4 hour or 24 hour generally the time window which we take. And in case of protectors, we generally give the drug, 1 hour or 2 hours prior to irradiation. And then we observe the survival. So 30 days survival. So that is called, we give generally a LD 130 dose, which is anywhere for different strains of mice. It generally varies between 7.5 gray to 9 gray. And then we observe the mice till 30 days. So all the death, as I said, like the death has started happening around 10 days and it gets over by 20 days. Maybe in this experiment, in some experiment, it may go on up to 24, 25 days and it starts from 12 days. So we have observed different period, but generally all the mice, they generally die. At LD 100 dose, they generally die by 25 days. So that is why the observation period is given up to 1 month, that is 30 days. Here we have, how we select when we are developing the drug. Then we need to find out that when we are giving the drug before, what should be the time window, and then whether, you need to find a target molecule, whether your drug is working. You have checked it at the cellular level. Now, when you are going to the animal, then you have to check whether your drug is working in the similar way or not. So here we have taken a molecule which is actually AKT signaling inducer. So AKT signaling is basically like what we did, we took a molecule which is inhibitor of the PTEN kinase. So if you inhibit the PTEN kinase, then AKT signaling, PTEN generally inhibits the, that is a phosphatase actually. So it dephosphorylates the phospho-AKT. So if you inhibit, then AKT will remain phosphorylated for a little longer, and then AKT signaling will take place in the cell, and that leads to actually the protection. So what we did here, you can see here in the Western blots, we have given the drug and 2 hours prior, and then after 2 hours we have taken PBMCs, blood cells, then GI cells, and then bone marrow, and then spleen, because these are the main targets, like PBMCs, bone marrow, and spleen of marker of hematopoietic, whether this signaling is taking place with this drug in hematopoietic system or not. And GI cells we have taken, and then run the Western blot, and you can see that phospho-AKT level has gone significantly high. There are three animal samples we have taken, and you can see the phospho-AKT level has gone significantly high as compared to pan-AKT. And then we are assured that we are inducing phospho-AKT signaling in the in vitro, and we are getting good radio protection at cellular level. So when we are giving it into the mice, this is inducing AKT signaling in different tissue of interest of us, and then probably it will be able to protect the animal also. And then when we expose to the radiation, you can see here, it gave around 60% protection in this animal. So not like 80%, 100%, which we got with other drug molecule, but this drug molecule also gave 60% protection. And this study was also recently published last year. So this is just an example that how basically we translate from a this radioprotective research, tissue protection, and the animal protection, how we translate from the in vitro level to in vivo level. So thank you very much. If Vibha ma'am want to add anything, or if there are any question, I am ready to take the question.
Yeah, so there are certain questions. So should I take Dr. Kale? Hello?
Yes, ma'am, please go ahead.
You can take, because
Okay.
I thought that
Ritu
madam is handling, so I was keeping quiet.
Okay. So Ritu, so there is one question that I think it is, can we determine LD50 values for cells in vitro? If yes, will the values be more or less than when compared to whole body irradiation? I think in vitro and then in vivo, there is some confusion. Are you going to take it, Anand?
Yes, ma'am. Yes, ma'am. So as I said, like some cells are very susceptible to radiation. So I am taking example of murine macrophagic cell, monocytic cells, that is called raw 264.7 cells. We use very frequently in our lab. So it is anywhere between 2 to 2.5 gray in different, if dose rate will vary, then its LD50 will also vary. I think somebody would have taken the class on this also, like dose and dose rate both are the critical determinant of the cell death. So if the dose, so anywhere between 2 to 2.5 gray, there is you will observe 50% cell death in these cells. However, when you go to a gut cell, like we use INT407 cell line, IC6 cell lines in our lab. So these cells, they generally show around 4 to 50% cell death at 4 to 5 gray. At 24 hour, I'm talking about. If you go on 48 hour, then this dose will further go down, right? So it depends at what time you're observing, and what dose rate is there, what dose is there. So there will be definitely difference between LD50 of, like, as I said, 2 gray cannot be the LD50 of any murine model. So the LD50 of any murine model will be not less than 7 gray or 7.5 gray. Like for this C57 strain, it is generally 7 to 7.5 gray. For some mice, white mice, maybe bulb C or strain A, it may be little lesser than that.
So Dr. Mohammed, Mr. Mohammed, did you get your answer, or you can ask yourself? Because we have enough time. If you want to ask anything, yeah, please ask what you want to ask.
Yes, ma'am. I've got my answer, ma'am. Thank you so much. Thank you so much, sir.
Yeah, and then you have written here LD50 values in cells in vitro, and then you are asking, your next question is talking about whole body irradiation.
Yes,
So
I just want to know whether the LD50 value will be more when compared to whole body radiation. Like we will isolate some cells and we are giving it radiation. So this radiation will be required more to achieve the LD50 value in cells, or will it be less in case of the cells?
Yeah, so that's what I told. It depends completely on cell system, what cell system you are taking. But generally, whole body LD50 will be higher as compared to individual cell, because you are exposing the cell completely naked. I mean, they are in the petri dish when you are exposing them. So, and they are directly getting exposed to the radiation. So definitely the damage will be higher. Damage will be higher, and LD50 will be lesser in case of cells as compared to whole body. However, you know, like in cells, it is a isolated system. So how in the animals, it may, like if I'm talking about macrophage, monocytes are very susceptible, lymphocytes are very susceptible. That will happen in 2 gray, 3 gray range. But if you'll go to macrophage, which is already differentiated, you will, I mean, macrophage will not die even at 20 gray. Right, 15 gray, 20 gray. So it depends completely on cell systems. Some cell systems are very resistant, some cells are very, susceptible. However, the whole body radiation is sum of this entire thing, right? So it depends on cells. So overall, you will get average when you are talking about an animal.
Okay, thank you.
Yeah, you cannot, I mean, generalize it. This is what I want to mean.
Okay.
Yeah, so there is one question from Chetan Shivrankar. Would you, Mr. Chetan, would you like to ask yourself? Okay, I'll read it. There was a slide comparing cell growth on giving radiation plus drug, radiation plus drug 2. Was the radiation dose same in all three? Should not there be a synergistic effect? I don't know which slide he's talking.
Which drug?
Means he wants to talk that, but I don't know whether you're talking about the protection, or at that time you were talking about syner.
Yeah, if you can tell me that which
I think
slide,
this
then
one will be there only, because here you have only one radiation. No, you have two radiations, two kind of radiation.
No, ma'am, I have given, this is 7.5 gray only, and then the drug was given prior to radiation. So, so the synergistic effect, matlab, see, there are different kind of drugs. So synergistic effect, we are talking about only when we are using drug as a sensitizer of the radiation. So the drug has to increase the effect of radiation, more killing. That is in case of cancer patients. Here we are trying to protect the tissues from radiation. So here it is not about, I mean, if suppose that the drug is acting other way, then there will be more death, there will be more cell death, there will be more, death will be observed in animals. However, these drugs are for protection. So there is nothing like synergistic effect here. So these are two different things. I mean, when we are treating cancer cells, then we need sensitizers of the radiation. They are different class of drugs. They will potentiate the effect of radiation. But here, these drugs are protecting the cells from radiation. These are normal tissue protection.
Yeah. So then another question is there. If any one of you don't get a satisfactory answer, please discuss with us. Otherwise, the whole process of this tutorial has no meaning. So you can please feel free to ask. So there is another question that, have you checked for the differential response of radioprotectors or mitigators in malignant or non-malignant cells? If yes, what kind of a response did you get? I think you can answer.
Yeah,
They
so
want,
yeah, yes, ma'am.
Go ahead.
Yeah. So what happens, like when we are talking about these drugs, see, when the scenarios which I talked, these patients, majority of these subjects, they may not be a cancer patient. So here, these molecules are developed from that perspective that it may induce proliferation. It may, it may protect the human or the animals from deleterious effects of radiation. However, these kind of drugs should be used. One market, or you can see one window where these drugs are generally given in cancer patients also during radiotherapy for protecting the normal tissue from the radiation. So in that case, these drugs should be used differentially. But majority of drugs which I talked about, they protect even cancer cells also from radiation. So that is why these drugs which I am talking about, they should not be used. They are not used in case of in cancer patients. What I'm talking about, this is a completely different window. See, here we are making this drug for any nuclear disaster and that kind of nuclear accident kind of scenarios.
Okay, and there is another question which says that what is the possible outcome in terms of lung fibrosis if immunomodulators are given along with the radiation to a lung cancer patient?
Yeah, generally, if you suppress, that is what observed even in case of COVID patients also. Basically, the steroids were given and so many immunomodulators were being given. It is basically to curb the cytokine storm which is taking place in lung. So of course, immunomodulators will work. So it should be given before this radiation pneumonitis sets in the lungs. So if it is given at that point of time, probably the outcome will be much better. The fibrosis may not develop. So that is why, like in case of, there is one, a lot of people use bleomycin, which is called as a radiomimetic drug. So bleomycin induced lung damage is also very well established. So people have seen that in mice, if you give bleomycin, they develop fibrosis, and after some time you will see in the same mice the fibrosis is gone. So they develop generally reversible fibrosis. So not in all the mice, but in some mice it will be gone. So probably it is because of the differential individual sensitivity to this and individual management of immune system in those animals.
Okay, there is one question, I think two questions. One somebody sent to me also, that how do we really do check the extent of damage through histopathology, how to do scoring, and what are the software available for doing the look at the tissue damage, and what software we should use. So that is what they just want, a little bit of information.
Yeah, so in histopathology, H&E staining gives the complete, generally the idea about, I have shown here. If you see here that this is a simple H&E staining, and you can see that if there is, you can take a help of pathologist to comment on this, to give their opinion on this, and otherwise also, which you see in the literature, you can see here very, lot of blue cells are there. They are gone completely. So, and then when you're treated with drug, you can see that they are there. So probably they are protected, or the drug is inducing proliferation. That further we need to dissect out. So this is suggestive of that the protection is there. But of course, there are certain software also comes, which which gives, I mean, some idea about how basically these, if you can see here in this, this is called Ashcroft score. So the lung, for the lung damage. So here you have to question this and that, and you can see in the in radiation, this score has gone significantly high, and the cells which were the mice which were treated with drug and then given radiation, this score has come down. So this score this is generally measured for analyzing the lung damage. So there are softwares also available which can I mean examine the images and then can give you the I mean the data about it.
Yeah, so next to it, I will, so I think there is, if there are, I saw Pragya, you can ask the question. You can unmute and ask the question.
Yes, ma'am. Sir, you have shown the results of macrocolony assay in one of your slides. So, sir, it is reported in some of the articles that after irradiation, the cells are kept in the incubator for half an hour or 1 hour or 2 hours before seeding, which will affect the clonogenic potential of the cell, as explained by Dr. Dwarkanath sir, due to the activation of repair machinery. So what should be the incubation duration that should be followed after irradiating the cell before seeding?
Yeah, that is called holding basically. So what happens, like generally there are different protocols. So there are two ways first to do macrocolony. The first thing is you irradiate as them as a I mean monolayer, and then in that case only liquid holding is possible. So what is there that I mean you hold them for some time, let's say 2 hours, 4 hours, and then you cut them and then plate them like 100 cells per PD or 200 cells per PD, depending on the control or radiation dose, and then you observe. So, but not every time, like when we do many experiments, we generally don't give holding time, because, and we see the holding time means you are not giving any growth stimulator. I mean, these media will have only the glucose as well as the medium supplement like HBSS media or PBS along with the glucose. So only basic very minimal media is given for holding, so that repair will take place, and then only you will give the growth stimulator, which is the serum complete growth media with serum, so the cell will start growing. So it depends, like generally in some experiments we give liquid holding holding time, and in majority of the experiments we generally don't give holding time. So how you will do, I mean, if it is post plating, then you will give exposure to monolayer cells, and the number of cells you will expose to radiation will be very less. Number of PDs you will expose to the radiation will be very less in this kind setting of experiments, and then you come back and then you dislodge those cells and then replate them as per the number. The second method is called pre-plating, so in which you already plate 200 cells or 300, 400 cells depending on the radiation dose or control group, and then you keep it for overnight for attachment, these cells, and you go morning and you irradiate. In this case, the number of PDs you will be exposing will be very large group, because for every group you will be minimally irradiating 3 PDs minimum. So so the number of PDs you will expose will be larger, and then you will bring them back and then keep again in the incubator for 7 days or whatever, depending on the doubling time of your cells.
Yes, sir. But sir, we don't do pre-plating. We seed the cells after irradiating them. So I was willing to know that for how long should we keep it in the incubator after irradiation? So that since
that you have to standardize. The good time is 4 hours actually. So you can give, I mean, this time you need to see through a Comet assay actually. So in Comet assay, you will be able to see that if suppose that in 2 hours your all the DNA repair is done. So in that case, you need to give 2 hour holding time, If suppose that you want to give 4 hours, because some damage will also be there, remain there after 2 hours, not the entire damage will be gone. So you can give 4-hour time. So depending on how much time you want to give. But if you want to take a buffer time that whether at 2 hours it is done or not, give 4-hour time, holding time, and then you do this post plating.
All right, sir. Thank you.
I can see 2 more people have. Yeah, so Palak, you are asking something.
Sir, I have a question regarding the protocol only. So can I suppose for the clonogenicity, can I see 300 and 600 cells and then irradiate and see the colony formation?
What is the plating efficiency of your cells?
Around 60%.
60%.
Yes,
and how much dose of radiation you are giving?
2 gray. And 4 gray, I am giving 4 radiation fractions, that is 2 gray, 4 gray, 6 gray, and 8 gray. And
Okay.
it's a single fraction dose.
Okay. So see, in your case, if you will be, it is plating efficiency 60%. So if you'll see 300 cells, it will form 180 colony, which will be very difficult to count.
Yes,
you are doing it in PD60 or PD35? 6-well plate?
35.
6-well plate. Okay.
Yes, sir.
So I don't think you should seed more than 100 cells, because in 100 cells, if you are getting 60 colonies, 60 colony itself is a big number in a PD35, right? So 100, not more than 100 cells you should seed for control. And for 2 gray, the cells which you are using is how susceptible they are. I mean, at 2 gray, you see very good number of good death or very minimal death.
No, sir, I have a question. Suppose in clonogenicity, we seed like 0.5 million cells, we radiate them and then we seed them in the 6-well plate, right?
Right.
But what I'm saying that instead of radiating my 0.5 million cells, I will first seed the cells in the 6-well plate and then radiate them.
Post-plating, pre-plating,
Yes,
you mean,
yes,
right?
yes,
No, that's
sir.
what I told. So in pre-plating, the number of cells will decide on 2 factors. The one thing, what is the plating efficiency, and and how sensitive. Suppose that at 2 gray, you are getting 50% cell death, right? So
Yes, sir.
so in 100 cells, your control is forming 60 colony. And at 2 gray, you will have 50% cell death. So your treated disc, which is like 2 gray exposed disc, they will form 30 colonies. So the number of the chances of error will be relatively higher. So here you should seed 200 cells. At 2 gray, you should seed 200 cells. At 4 gray, you should seed anywhere between 400 to 600 cells. And at 8 gray, you should seed around 2000 cells. So in 2000 cells, you will see hardly 10-20 colonies will be formed at 8 gray. Right?
So depending upon the plating efficiency and the susceptibility of these cells to the radiation, I should proceed with the seeding.
Right.
Okay.
At that given dose, it should have, let's say, 30 to 40 colonies minimum. At high doses of radiation, you will see only very few colonies are formed. So if the colony numbers will be very less, the error in your data will be very high, right? So that is why I'm telling, use that number of cells which forms at least 30-40 colonies. At control level, 100 cells will be sufficient.
And on what basis I should decide? Like yesterday, Dwarkanath sir, he was telling in the clonogenic assay only, he plated the plate with the agar, the underlining agar, with the nutrient, and then the cells, and then on top of it there was one more agar layer. So how should I decide, like whether should I do the agar plating or not?
I think he would have told you about suspension cells when you are forming the colony of suspension cells. So if the cells are adherent, you don't have to use agar because they will adhere. I mean, the image which I am showing you currently, they are adherent cells, no agar used. I mean, they will attach to this petri dish and they will form colony on the petri dish without any agar.
But few of the
You go ahead.
But few of the publications, they have shown that for the NIH3T3 cells, fibroblast cells, they have used that method as well.
Agar.
Yes,
See,
sir.
agar, some people, they use agar to avoid basically forming of satellite colonies. You know, by putting agar, what happens that the cell will not detach from here and will form a neighboring colony. So you don't know. So principally that cell, suppose that the colony A, there are A, B, C, D, 5 colonies in any petri dish. So if some cell get detached from A and it form a colony F there. So you will misinterpret. It is basically the interpretation should be that the colony A should be only count. A and F should be count as one colony. But when you will see, you can see here also some very small colonies are there in this radiation group. Okay. So you really don't know this has come from parental colony, or this is actually a colony where the cell number has not grown up to 50 or more. Generally, we count for six doublings to say that this cell has clonogenic potential, right? So to avoid the satellite colony, sometimes agar is also used.
Okay, thank you, sir.
But you need to find out a window when the cell starts detaching and forming a satellite colony. So don't wait for very long. I mean, don't just keep it for 20 days and you forget. So after 4 or 5 days, you should keep observing that when the colony size has grown sufficiently, you terminate the assay.
Okay, thank you, sir.
So is there any more question from anybody? So I don't see any other question. So I would like to, Sridevi Pathak again wants to ask. Yes, Dr. Yes, Sridevi, tell. Yeah, please unmute and talk to us. Yeah.
Sir, while irradiating the cells, we are directly irradiating the cells, or do we use, from one of my friends from medical physics field, I got to know they use phantom to irradiate the cells. So do you have any idea about it?
What they used, ma'am?
Phantom, she said.
Phantom, okay. I don't know exactly about it, but are they collimators and all?
She couldn't explain to me the concept of phantom. That was the problem. Okay. But they have some box kind of a thing in which they keep the petri dish before irradiating the cells.
Okay.
And the matrix is plain water only, I guess.
Okay. So, ma'am, actually, these are different machines, and which in these different machines, the setting for exposure is very different. So I think medical physics people can give you better idea, because whatever machine we are using, there should be homogeneity of the radiation. It should not happen at some place radiation is coming more and at some place radiation is coming less. So so how we do that, we use teletherapy machine. So, and we make a 35 by 35 area. That area will be illuminated by light. So you know where is the boundary. So we keep all the petri dishes in that area, and then we switch on the machine. So it will expose all those petri dishes directly on the platform. We keep just beneath the wherever this exposure machine expose in that exposure area, and then it exposes and all the dishes. So there should be a source to platform distance also, that is called SSD. So that SSD determines that how much what is the dose rate. So by changing this SSD, you can change the dose rate also. So these are done by generally medical physics people. They do the dosimetry also, like as I said, in that 35 by 35 area, they do dosimetry, and then they confirm that in this 35 by 35 area, everywhere the amount of radiation dose is coming is similar to. So that is how we are, we are okay with it that wherever I keep my dishes, the equal amount of radiation will go. Otherwise, suppose that if I am exposing 3 dishes of for 2 gray, so dish A may get some other amount of radiation, B may get some other amount of radiation, and C may get something else. So that is why this homogeneity is also important.
Okay. Thank you, sir.
Yes, sir.
So thank you very much, Anant. And I would like to ask both of my chairs, Dr. Ritu and Pragya, please make Dr. Ritu co-host. She has rejoined us.
Okay.
So I think she was out.
Here only, ma'am. I've just, because of the bandwidth, I've switched off.
I know. So I just want Dr. Ritu and Dr. Kale both. First of all, I would like to ask Dr. Ritu to share her research area and where she is working, because so this is a platform where the students would like to know the like people who are working researchers, and they may benefit by your experience. So if you tell us about your research area, then it will be good there.
Okay, so hello everyone. I have been in the introduction itself, I think Antara spoke a lot about my research areas. So I basically deal with hematopoietic malignancies, and I'm a hemato-oncopathologist. So essentially, most of my clinical work deals with the diagnostic aspects of these cancers, their differential diagnosis from other benign as well as other malignant entities. We also deal with a lot of data pertaining to prognostications of these disorders, and also we do the therapeutic monitoring, which essentially means that we are looking at minimal or the measurable residual disease. And in the process of doing that, we are using a large number of latest state-of-art technologies. Like we use the multiparametric flow cytometry, and now because the measurable residual disease requires detection of very, very low levels of the disease in the milieu of normal hematopoietic elements. So we use next generation flow cytometry, something similar to next generation sequencing. So that's an area where our laboratory has immense experience in Excels. And if any of you are interested in really going into that area, you may contact us. Another area is that because essentially, if you look at the development of all the sequencing technologies and how they come into the clinical foray, a lot of work has been done in hematopoietic malignancies, because these cells are already in suspension and easy to access. So our laboratories deal with a large number of molecular biology technologies, including Sanger sequencing, next generation sequencing, the digital MLPA, the normal Sanger-based MLPA, all types of PCR reactions, and much more. So that's in brief about the various technologies in the cancers that we deal with. And apart from that, there are a lot of other aspects. I'm sure if anybody is interested in to work on hematopoietic malignancies, we can discuss them.
Okay, thank you. It was nice for that. And sir, Professor Kale, are you here?
Yeah, thank you, madam. I'm not active researcher.
Yeah, but you can always share your
because now I have not in the laboratory or near the working table also. After superannuation, I shifted to Pune, and I thought that and there somehow because of earlier involvement in some administration policy decisions, so I was a little bit away, and then I discontinued the active research working as such. But however, during my tenure as a teacher in the life sciences, I worked in the radiation damage and its mechanism, and try to explain on the basis of the chemistry, particularly the free radical biology, and try to understand how we can explain the radiation damage. And along with that one, I have also worked with the protection sensitization, and very interestingly, using the many radio protector and sensitizers. But I come to conclusion that there is no watertight compartment between the protector and sensitizer for the radiation therapy, and for that matter in other places also as a antioxidant. because basically protectors are the electron donor, and sensitizers or toxic chemicals generally are the electron acceptors. But this property depends on the capacity to donate and capacity to withdraw the electron. But this gets changed due to the chemical surrounding, and from that, I try to explain that there is no watertight compartment between the antioxidant and oxidant or the radioprotection sensor. They can behave accordingly, depend on the chemical surrounding, and that I was able to give this one because this is a uric chemical reaction is under the control of chemical thermodynamics and kinetics, and so how this variation takes place, that's more important. I try to give explain most of the behavior of the radioprotector or antioxidant or sensitizers or the toxic chemicals on that basis.
Yes, sir. I'm in a meeting. I'll call you back.
Apart from that one, I also work in environmental carcinogenesis, that is chemo prevention, and we use several plants products, and we have shown that how this work also. And there also we have seen instead of taking the single the principal product, it may not necessarily useful. It may be good for the pharmacist or the industrialist to produce some product and sell in the market and make money. But stoichiometry is also equally important because the chemicals are working along with other chemicals. That's more important. Therefore, the stoichiometry also we try to explain that the entire fraction, the compound fractionated either in aqueous or non-aqueous medium. They are better for the chemo prevention. But anyway, to explain the effect of the certain plant product, we need a pure chemical, and on that basis we try to explain how they do as a work as the protective agents, or they work as a sensitizing or toxic agents. This was generally overall. Along with that, I also work in the case of dose rate and total dose. And this is a very interesting area. If you change the dose rate and use the same dose, the effects are different. And we try to explain that also, dose rate, and also another area I did a little bit work. So these are some of the areas. But if somebody wanted my help on theoretical level or something like that, I'm always there. And I have my email address with Vibhaji, and I think anybody can contact me. And I think I'll be more than happy if I can help them some or other way. Thank you very much, Professor Vibhaji, for giving me this opportunity to explain my area of research, what I did earlier. And there are also other few things I have done, but I think this is enough for today's thing.
So over to both the chairs, so they can close, Ritu or Dr. Kal.
Yeah, I think I started. I think I'll request Ritu Madam to close. Yeah.
This was a wonderful session, starting from Professor Dwarkanath and then ending with Dr. Anant and Dr. Vibha. I'm sure all the students and including the faculty members have also taken this opportunity to freshen up their knowledge and education. So thank you, everyone, and thank you, the GIAN initiative and Professor Vibha Tandon for such a wonderful, fantastic program. Thank you all, and now we close the session for lunch, for a short lunch break. And we reassemble at 2?
Yeah.
2 o'clock.
Yeah. Pragya, are you speaking something? Yeah.
Yeah, we are thankful to the respected chairpersons for their excellent conduct and lending us time to agreeing on chairing the session. And we are also thankful to the speakers, Professor Anand Narayan Bhatt sir, for elaborately explaining the biological consequences of radiation through his published articles, and Dr. Dwarkanath sir for very well explaining the DNA repair mechanism and radiation-induced cell death. Now we'll be moving towards lunch break, and we'll be back at 2 p.m. to proceed with today's program. Thank you.
Thank you, everybody. Thank you, Anant, and thank you, Ritu and Professor Kale.
Oh, thank you, Madam. It's
Okay,
honor and privilege. Yeah.
Okay, bye-bye. Thank you.
Bye.
So thanks for all. Okay. So I think Dr. Kale is here and Dr. Amitabh is also here. Pragya, we have to wait for some minute. Let participants should come. I saw Dr. Amitabh also joined us. Yeah, he's here.
Yes, ma'am, I'm here. Good afternoon.
Yeah, sorry, I by mistake I wrote 2:30 in the morning.
Okay, no problem.
So we are just waiting for some more people to join and then we'll start.
Hello, ma'am.
Hello, welcome, Pooja.
How are you, ma'am?
Fine. अमिताभ, मैंने ना आपको कुछ पूजा के अवार्ड्स वगैरह भेजे हैं। I think Pragya, we should start the session, and Dr. Pooja, Dr. Amitabh, and all other participants, Professor Kale, and Dr. Akhilesh Verma. So Pooja has another teacher who is going to look at Pooja. Right? So
hello Pooja, how are you?
Hello, sir. How are you? I'm good.
Good, good, good. Good to see you.
Thank you, sir. Same here.
she is our speaker today, our expert. Okay, Professor Kale, are you here, sir?
Yes, I'm here. I'm here.
Okay. Yeah.
So happy to see our students on the
Yeah.
forum. It's a very, it's a proud moment for all of us.
So
अखिलेश जी
she
कैसे हैं?
Thank you so much, sir. Thank you.
अखिलेश जी कैसे हैं?
Yeah, I'm good. I'm good. आप बताइए कैसे हैं?
बिल्कुल ठीक. After long
After a
time.
long time,
Yes,
we
yes.
are meeting on this
Yeah.
virtual forum.
Right.
Pooja, Pooja, do you know Dr. Ramesh Chandra is also here?
Yes, ma'am. I've seen, but I can't. His video was on, but I
he
not.
will open his screen. Don't worry. His screen was open.
So Kale sir, is any in future are you coming to Delhi?
Not immediately.
Okay.
Okay, so Amitabh is also there. So Pragya, you can start now.
So welcome back, everyone. So I hope we all are back with all the excitement to learn about the advancements in science and technology through this program. So without further delay, let's start our next session. This session will be chaired by Dr. Amitabh Singh, in charge of Pediatrics, Hematology, Oncology Division, Safdarjung Hospital, New Delhi. Dr. Amitabh is an associate professor at Vardhaman Mahavir Medical College and Safdarjung Hospital and has his expertise in pediatrics and hemato-oncology. And Professor R.K. Kale will be joining with us again to chair this session with Dr. Amitabh. Professor R.K. Kale is...
No need, because morning you are done.
All right. Sir.
Yeah, we can save time.
All right. So I request the chairpersons to take hold of the session. Thank you.
Hello.
Yes, sir. You are audible.
Yeah, Dr. Amitabh ji.
Yes, sir.
Please go ahead. I think you initiate. Yeah.
Okay, sir. Thank you for inviting me, and it's my pleasure to introduce our eminent speaker today, Dr. Pooja, who is Scientist F and Joint Director Head, Division of Cyclotron and Radiopharmaceutical Sciences, Institute of Nuclear Medicine and Allied Sciences. She has a lot of good publications, or I should say she has more than 94 publications with over 900 citations, and she also has long list of awards, which was I will just mention few, and then we'll start the session, and we are eagerly waiting to hear and learn from Dr. Pooja. So she was the Laboratory Scientist of the Year, DRDO, for 2013 and 14, and she has a lot of best paper awards, and her area of work includes diagnostic imaging, molecular imaging, PET, and all the theranostics, which we are going to hear. So she is the best person to educate us more about the theranostics. So I request her to start the session. Thank you.
Thank you so much, Dr. Amitabh, for such a modest introduction. So I'm very thankful to Professor Vibha Tandon for inviting me, and I'm very delighted to be a part of GIAN. And also I am taking this platform. I would like to thank her because she is the one who has sent me to Institute of Nuclear Medicine and Light Sciences for my dissertation work. And then she used to say that now I have sent you there, and now you'll never come back from there. So thank you so much, ma'am, for this nice interview that you have inculcated in me, that the research. She's my guru, first guru in MSc. So thank you so much, ma'am. So with this, I would like to share my screen. I think.
Are you able to see my screen now?
Partially.
And now?
Now it's okay.
It's okay. It's in the slideshow now.
It's not in slideshow.
I've done that in slideshow. Should I?
Pooja, there is a issue. If you, what you have to do, that you have to go
Entire.
Entire is fine. Yeah, you have to restart.
Now, is it visible?
Again, वो नहीं, same ही है. तुम्हारे नीचे slide show नहीं आ रहे थे? Can CIS people can say help something? कोई बता सकता है? हाँ, fine.
Again, it's done.
Yes, yes.
So, good afternoon, everyone. And as my introduction has already been done. So I'm associated with Institute of Nuclear Medicine and Allied Sciences, which is a center of advanced molecular imaging. So here, what we generally do is we synthesize the radiopharmaceuticals for targeted imaging and therapy of cancer, and also for neurocognitive disorders, and also for neuroendocrine tumors. So if I go to the imaging modalities that we have today is the SPECT, which is a single photon emission computed tomography. PET imaging, that is the positron emission tomography, MRI, molecular magnetic resonance imaging, optical imaging, and therapy, and how it works. Like imaging modalities, if you look at the SPECT and PET, these are heavily dependent on exogenous tracer. So the development of a personalized approach in healthcare is motivated by the call for improved efficiency, safety, quality of treatment. Early diagnosis plays an important role and is one of the major factors that reduces the mortality in cancer management. The remarkable achievements in genomics, proteomics, and biotechnology have contributed drastically to the emergence of individualized diagnosis and therapy on a molecular level. So high-resolution molecular imaging is one of the fastest growing imaging procedure worldwide and is considered as a major breakthrough that strongly contributes to the realization of personalized medicine. Nuclear medicine includes such technologies such as PET, SPECT, and uses gamma and positron emitters radioisotopes for generation of the signals that results in the whole body scan in a single examination. So we talk about SPECT as a classical SPECT. Only these modalities were available. Only SPECT was there, where you can only do the SPECT imaging, and only PET imaging was there, where you can. Why I'm saying only PET imaging, because now is the era of hyphenated imaging or the hybrid imaging, where you can see that we can mix or we can hybridize two modalities together, like. SPECT could be hybridized with the CT to complete the picture of the physiological, functional, anatomical details. In the same way, PET is also can be hybridized with CT for the anatomical details. Why they are being hybridized in time now? Because now if you see what PET can image at a molecular level, means you can see what is happening inside the body at the molecular level. And the same way, SPECT is also, you can read the molecular detection in SPECT. But in CT, if you look at the CT, you can see the anatomical details very nicely, but you're not able to apprehend the molecular fluctuations in the receptor that is happening inside the body. What are the biochemical pathways that are disturbed that cannot be seen by CT? And if I talk about the conventional MRI, in conventional MRI also, we are not able to see what at the molecular level what is happening. But yes, MRI provides very good anatomical details and somehow, somewhere, physiological details also. So if you talk about the non-invasive imaging, if you see the CT, I have distributed this into 4 parameters like anatomy, physiology, metabolism, and molecular. CT could show anatomy and to a little bit of the physiology. Ultrasound and CT, they say we have the same information that we decipher from CT and ultrasound. MRI, yes, anatomy, physiology, metabolism, and to some extent molecular level. If we do the functional MRI or the bold MRI, we are able to see some process, molecular process at the biochemical level or the molecular level. If you see about PET, if I look at PET imaging or the SPECT imaging, I can little bit have the idea of the physiology and the metabolism, but I definitely have the clear, very vivid picture of what is happening at the molecular level, at the biochemical level, what receptor of fluctuation are going on, even if it is of the cancer or the neurodiagnostics or the neuroendocrine tumor or any kind of tumor or some kind of receptor fluctuation is there. So I'll be able to detect it by PET and SPECT. Optical imaging is also deciphered at the molecular level, but it is not very deep-seated. So deep-seated tumors or the cancer will not be able to decipher from the optical imaging. So clinically proven 2 techniques like PET and SPECT are there, where you can observe the molecular imaging or the molecular processes, which allow the assessment of the pathological site in a receptor level, at the receptor level. So coming to the, because I have included theranostic agents for targeted therapy and diagnosis. So how we reach to a clinical level, developing a molecule which could be utilized as a molecule for diagnosis as well as therapy. So first of all, we design a molecular targeting system. It is designed about the pathological site that we have, that we want to diagnose. Maybe cancer, maybe neuroendocrine tumors, maybe cognitive disorders. So what kind of target we are choosing? It could be receptor, it could be enzyme, it could be a transporter. So depending on the target site, we design our molecule. And then to make it radioactive, we need to establish a synthetic procedure where we can incorporate the radioisotope inside the construct of the molecular, the entity that we have designed for the imaging and therapy. So after that, we do the cytotoxicity profiling of the prostatic groups, as well as the conjugates, as well as the radioconjugates. So we need to see how best it is binding to its receptor or the target of interest. For example, if I'm developing a molecule for acetylcholinesterase enzyme, so I have taken tacrine-based moieties, and now if I modify to such an extent where I'm incorporating a radioisotope or radionuclide inside the entity in the molecular construct of the tacrine, so I have to see that it does not compromise its binding affinity towards this target. Since it's a tracer technique, the binding should be in nanomolar to picomolar range, because what we inject in a human subject or an animal subject is a picomolar quantity of the radioisotope that we track down or trace down so that it does not have any biological or therapeutic response. So a question comes to the mind: if you are talking in tracer technique in terms of picomolar range or nanomolar range, how we are going to do the therapy when I'm saying that there will be no biological response as such by the tracer molecule. So you have already learned a lot about the radiation biology effect of the radiation. So that we exploit here for the therapeutic application. We take the radioisotope, which emits alpha, which emits beta, which have high linear energy transfer, those kind of radioisotope will be taken for therapeutic application. But the concentration that we'll be using will be in the tracer quantity that will be injected for the targeted therapy. So after completing this preclinical evaluation on both like pharmacokinetics and pharmacodynamics profiling, after doing that, we go on to the preparation of the animal models, having the substantial amount of receptor concentration that we are looking at. So to assess that, we do animal PET spec CT dynamic studies and biodistribution studies. And we find out that what is the clearance pattern, what is the how it is rapidly going to the target site, how it is the background is clearing rapidly, so that we get a very good signal to noise ratio when we are doing the acquisition part. So after establishing all these parameters in vitro, in vivo, we move on to the development of the dossier for establishing a human protocol. And after ethical clearance, we could reach to a stage where we can inject these radioisotope-based radiopharmaceuticals or for theranostic agents for diagnosis and therapy. So talking about our center at DCRS, if I talk about the productivity of DCRS since 2007, so we have developed various range of imaging probes for targeted imaging at en masse. We are also into the production of radioisotopes, different radioisotopes, which are PET radioisotope, which are cyclotron based, and generator based radioisotope also we have produced. And we have also transferred technology for gallium-68 radio labeling in form of a workshop that we have conducted in 2013. And we are now beginning to produce maybe therapeutic radioisotope with the help of the cyclotron. Now coming to the molecular targeting, what could be imaged with PET and SPECT? Whatever biochemical pathways are there, like glucose metabolism. You have heard about a lot of lectures on 2DG. When Professor Rath also, very nicely explained about the 2DG mechanism and everything. So 2DG goes to the various oncological, it's a radiosensitizer. It helps in, it is transported through Bluetooth transporter. In the same way, same mechanism would be employed when we are developing a molecular targeting agent for imaging. So fluorodeoxyglucose is nothing but when you see 2-deoxyglucose, where you are removing the OH or the oxy group from the glucose, it becomes 2DG. And when I'm replacing OH or the oxy group to the fluorine group, which are both are bioisosteres, it does not changes its integrity of the molecule. And FDG also, in a similar fashion or a similar way, it goes to the targeted site on its target, targeting the cancer, inflammation, infection. So it has a role and multiple roles for FDG are there. So it goes in everywhere, inflammation, infection, onco like cancer, and so on. So next comes the receptor or enzyme imaging. That is a very, very specific or targeted, I would say, where you can also do the receptor quantification when you do the imaging. It becomes like the amino acid transport. You can track down the amino acid transport in various cancer or oncology. In oncology, various type of transporters are required by the cancer cells, like large amino acid transporter to transport methionine, to transport dopa, phenylalanine. So there are different amino acid transporter that is utilized for by the cancer cells. And the ratio on that amino acid transporters are also very highly expressed, or they have the substantial amount of receptor concentration or the transporter concentration on them. So we can track down in a very nice way, having a very high contrast ratio if we use the amino acid transport. So here, large amino acid transporter are L-type amino acid transporter, LAT1, to be very precise, is overexpressed in most of the proliferating tumors And LAT1 would be targeted by methionine, dopamine, and tyrosine. There are a lot of amino acids which we can utilize to make them very specific to the target. After that comes the nucleotide imaging, where thymidine could be exploited for the cell proliferation. So for that also, there are mechanisms where we can incorporate the radioisotope into the oligonucleotide or the thymidine precursor. The most common example is the fluoro-L-thymidine, where thymidine is labeled with fluoro-18. We can also target the lipid metabolism very nicely in tumors. As it is rapidly growing, it requires a lot of choline kinase enzyme is expressed. So enzyme targeting can be done with the help of choline. Now, what are the application in nuclear medicine of these radioisotopes? We are talking about the radio. There are three different things are there, like radiopharmaceutical. Radiopharmaceutical is made up of a pharmacophore group, which we are targeting for a specific kind of a receptor on an enzyme. Then we have the radioisotope as such. So radioisotope is the most important part of any theranostic agent. So if we are looking for the imaging application, we have the gamma or positron emitting radioisotope, namely technetium-99m, indium-111. Now fluorine-18, carbon-11, copper-64. These are the positron emitters. So it can be easily utilized or exploited for visualization of a biological process like cancer, myocardial perfusion agents. The very good example for myocardial perfusion agent is O15. That also we produce here by the cyclotron. Now for therapy, there are particle emitters which have high linear energy transfer, and which includes alpha, beta, conversion of auxiliary electrons also. Rhenium-188, holmium, strontium, yttrium-90, bismuth, actinium, and iodine-131. So these are the most commonly used therapeutic radioisotope in clinical nuclear medicine. Now coming to the PET imaging probes. So what is PET? Positron emission tomography. How positron is emitted from a radioisotope, how it gets into the signal that could be transformed into a useful information. So here, the annihilation is the process by which the radioisotope, which is positron emitter, it reacts with the matter, it reacts with electron in the matter, and then annihilation takes place, wherein the two gamma photons are ejected in the opposite direction. To catch hold of these signals, coincidence detectors are required to get the signal where the signal is coming from. So it utilizes coincidence detectors. This is, you can see that in 1950, Gordon Brownell suggested that coincidence detection could increase both the resolution and sensitivity of nuclear imaging of brain tumors. So a simple scanner, positron scanner, was made, quickly designed and built, having two opposed sodium iodide detectors, and it was used to take the first image of the coincidence scanner. Now, if you see the modern day detectors, they are aligned in the 360 degree. They're aligned all over the ring. And then, so that the coincidence photons that are there, the signal is cached, and then images, after image reconstruction, 2D or 3D image could be obtained of the radio tracer concentration where it has got accumulated. Now, clinical PET tracer, we work on the clinical PET tracer that has been used in oncology at mass. So we are the one who are, designed methionine-based, choline-based, tyrosine-based, dopamine-based hamidine-based radiopharmaceutical, which have been used in clinics at our center. Now coming to the most conventional fluorodeoxyglucose, which has been utilized in oncology. The accumulation of FDG is based on enhanced glycolysis, which has often been associated with the growth rate and the malignancy potential of the tumor. So what it is, now also it is being utilized for staging and restaging the malignancies, evaluating the extent of disease of known malignancies. So if you have a higher SUV, standard uptake value of FDG, it is considered as malignant. So it is also utilized as a tool for differentiating benign from malignant lesions, and also selecting tumor region for biopsy guidance. Where it is highly proliferated or glycolysis is the SUV region is very high. From there, biopsy, a sample for biopsy could be taken. So pre-surgical planning could be done using FDG scan, but it won't be in itself complete without the presence of CT. So PET CT is done for the pre-surgical planning so that you have the coordinates for the tumor where it is located. It is also helpful in radiotherapy planning with therapeutic and palliative intent. So how much the chemotherapy is working, how much the other therapies are working or the hormonal therapy is working, FDG becomes a tool to assess the therapeutic or the radiotherapy planning with that, with the FDG. But there's still limitation of FDG. It is non-specific. It goes to infection, it goes to inflammation, it goes to metastasis, it goes to cancer. So how will you differentiate if FDG goes? How will you identify that, yes, if there is a FDG uptake, SUV level is very high, but now on the biopsy, it does not turn to be, very malignant, and it could be, without surgery, it could be treated through a chemotherapeutic agent. How will you know that? So that is very, very tricky when you use FDG for that application. So what we have done is we have introduced amino acid based carbon level labeled PET pharmaceutical, where we have labeled, we have tried to label methionine with carbon 11 at our center, where we have produced when you do the proton bombardment on nitrogen and oxygen, which is a balance of 99% nitrogen and some bit as a carrier. For carrier, we use 0.5% of the oxygen. So we are able to produce carbon 11 CO2. That is a carbon dioxide, which is radioactive in nature, and it is in gaseous form. So that gaseous form we have to trap down on the nickel shimalite column that is there in the module, where you can at 350 degree would convert carbon CO2 into methane. So methane is converted again, it is highly radioactive and it is gaseous form. Now this methane is converted into methyl iodide, which becomes an alkylating agent. And how we do it, it is being done at 750 degrees furnace. You will see it's done at 750 degrees furnace, where iodine is sublimed, and then it is reacted with the carbon 11 CO2 with methane, which is highly reactive in nature. So when we get this carbon 11 methyl iodide, this becomes the alkylating agent, which could be utilized to label any biomarker of interest, not only methionine. It can be utilized to radio label any of the biomarker of interest that is there to track down the other physiological or pathological conditions. So here we have utilized this technique, or carbon 11 methionine has been extensively used in detecting the low-grade and high-grade gliomas. So there is no diagnostic dilemma. If you see the images of the carbon 11 methionine compared to the FDG, so you could appreciate that carbon 11 methionine uptake would be seen very vividly in the scan A, whereas if you see the FDG, where the tumor, it is showing the tumor recurrence, And in FDG scan, the surgical procedure that was done on this patient, there is nothing inside this, but it is not able to give the information about the recurrence of the tumor. So with the help of this carbon-11 methionine modality, we can definitely, it is based on the LAT1, and we can do the grading. Grading the malignancy is very nicely done with this radiotracer. Delineation is very nicely done with carbon-11 methionine, which is a proliferating tumor. All the cells that are active could very well be seen on this carbon-11 methionine scan. It is very helpful in detecting the radiation necrosis versus recurrence. You will see this is a conventional MRI which was done on a patient, which is showing some abnormalities on the scan of MRI. So these are the portion that looks like tumor, but what in reality, when this patient was subjected to carbon-11 methionine, so no uptake was seen. So no proliferating tumor or proliferating tumor was seen. So it was reported that this, the conventional MRI, whatever abnormality was seen was due to the radiation necrosis. This tool is also, like carbon-11 methionine, is also very helpful in prediction of prognosis and evaluation of response to treatment. So when you see the grading and prognosis, prediction of prognosis, if you see the grade of malignancy, if the uptake is very high, so oligodendroglioma, which is WHO grade 3, the uptake is quite good in this case. In oligodendroglioma grade 2, the uptake is 2.34. So according, this is the SUV standard uptake value that that is being shown here. So according to this, we can also determine we can do the grading of the malignancy using carbon-11 methionine. So carbon-11 methionine, again, it has a limitation. Now you'll be thinking that it's a very good tracer. It could delineate, it could do the prognostication, it could do the grading of the malignancy. And now I'm saying there is a limitation. Yes, there is a limitation. Not because of the precursor, it is because of the radioisotope that we use. It's a carbon-11. And carbon-11 has a very short half-life. It's only about 20 minutes. So this 20 minutes half-life is the only limited use or the constraint that we have with this carbon-11 methionine radiotracer. To overcome that, we have again synthesized or incorporated fluorine-18, which has a 110 minutes of half-life to do the longitudinal studies in the patient. So we have introduced fluorine-18 ethyl L-tyrosine that we have made, which is called FET-PET. So this is the dynamic F18 examination shows high diagnostic accuracy in patient with suspected tumor progression or recurrence. It can also differentiate recurrent brain tumor from pseudo-progression and radiation necrosis. And how it is more, better than carbon-11 methionine or fluorine-18 FDG is that it clearly delineates tumor from inflammation. Carbon-11 methionine also has some propensity towards reaching the inflammation, but in case of fluoroethyl tyrosine, it only goes to the tumor and delineates the tumor from inflammation. So this is the advantage of using F18, F18. So now you can see the PET MRI. I've talked about PET CT. You can hybridize today. We are hybridizing PET CT, PET with CT, PET with the SPECT with CT. Now the PET MRI is also possible, and PET MRI cameras is also there in India at Apollo Hospital, and one has come in Nimhans. So this scan was taken, like we have sent this fluoroethyl tyrosine at Apollo, and this was conducted at the Apollo Center. Now you'll be able to appreciate the uptake and delineation of the tumor. You can find out the coordinates for the surgery of this anaplastic oligodendroglioma. Now coming to the F-DOPA, which is dopamine, which is also labeled with fluorinating, to evaluate the dopaminergic system functioning in brain tumors and neuroendocrine tumors. So when we talk about the Parkinson brain, so we have this basal ganglia, which is, you can show a good uptake of F-DOPA, which is fluorinating dopamine, very nicely uptake. So we can say that the receptor quantification that we have done is a normal receptor quantification on a normal healthy volunteer. But when we have seen the case, this is the case of the Parkinson, wherein the basal ganglia show very less uptake, showing that the D2 receptors or the receptors here are really depleted due to the Parkinson. So the extent of Parkinson disease could also be calculated from F-DOPA scan. So it used to investigate the activity of aromatic large amino acid decarboxylase to evaluate the dopaminergic system functioning in brain tumors and neuroendocrine tumors. So at our center, this particular molecule has also been investigated in detecting the gliomas, because this is also go with the large amino acid transporter. So high-grade gliomas could also be seen on this scan. So it is helpful in detecting primary, metastatic, and recurrent brain tumors, delineation of the tumor volume, and it also determined the proliferative activities and rhythm. Now coming to the another agent, that is fluoromizonidazole, PET imaging agent for tumor hypoxia. This is very, very useful in FDG PET non-responders and also response to radiotherapy and chemotherapy. It is a marker of perfusion, and it helps in management of the cancer. So how F-MISO scan is very, very helpful in hypoxic cell. F-MISO is trapped, which is the basis for the use of this tracer to measure hypoxia. Now coming to the another fluorinating derivative or the radioisotope labeled tumor-specific PET tracer, that is the fluorothymidine. Fluorothymidine activates in proliferating cells where it indicates the activity of the enzyme thymidine kinase. Now, it is suitable for monitoring how tumors respond to cytostatic therapy. So there are a lot of modalities which can be combined together to get a good therapeutic response management of cancer. It also has a positive role to play in predicting therapy response, especially in brain, lung, and breast cancer. Now coming to the lipid metabolism, to study the lipid metabolism, how we have designed the molecule and how we have labeled fluorinating has used successfully or assessed clinically significant prostate cancer. So F18 choline PET-CT has augmented the diagnostic imaging spectrum for the assessment of relapsing prostate cancer. So for F18 choline PET-CT in detection of bone metastasis in prostate cancer are 74%, 99%, and 85%. And it is superior to bone scintigraphy for the early detection of metastatic bone disease due to detection of bone marrow metastasis. So for bone metastasis, there is another compound which is based on phosphonate, which is labeled with or maybe sodium fluoride, which is fluorinating, could also be directly injected into the patient to get the skeletal scanning. But in this scan, diagnosis and follow-up and therapy and follow-up could also be done at the same time. So now coming to the theranostics. Now we were talking about the PET imaging probes. How PET imaging probes could be utilized as a theranostic agents. And what are these theranostic agents? Theranostics in its broader meaning embraces diagnostic methods conducted for the prediction of efficacy of specific therapeutic intervention on an individual basis, as well as for monitoring the response for the treatment. So here, a drug that would comprises both diagnostic and therapeutic function is called the theranostic agent. in a very layman term. If we define theranostic, what is theranostic? It becomes that it should be able to do the theranostics, therapy, and as well as the diagnostics. But in real scenario, is it there like any system which comprises of both? So how it is being done? Like, according to the definition, it seems that if we give the theranostic agent to a patient, it will simultaneously do therapeutics and diagnostics. So, but this is not the case. The radiopharmaceutical that we design for diagnosis could be utilized for therapeutic application by swapping the radioisotope of interest. Like here, if you see gallium-68, scandium-44, these two are positron emitters. And if I go on lutetium-177 to yttrium-90, these are therapeutic radioisotopes. So these radioisotopes, these 4 radioisotopes, could be tagged very nicely on DOTA-TATE. DOTA is the macrocyclic system which is attached to the peptide for detection of the gastroenteropancreatic neuroendocrine tumors, GPNETs. So for that, a system is designed where you can see the chelator. This is the chelator and this is the peptide. So this chelator could be utilized to bind gallium-68, scandium-44, lutetium-177, and yttrium-90. So theranostics for neuroendocrine cancer is defined as a find and destroy mission. So once we diagnose it with gallium-68, DOTA-TATE, when we see that there is accumulation of gallium, DOTA-TATE in the neuroendocrine tumor, then we plan the therapy with lutetium-177 or yttrium-90, depending on the tumor size. So this is being done, which is also called peptide-based radiotherapy, peptide receptor radiotherapy. So basically what we intend to do is the high value of molecular imaging lies in the early detection, staging, therapy selection and planning, as well as follow-up. The in vivo visualization and quantification of the biological process underlying the disease are the characteristics of nuclear imaging that provides information for the staging, lesion delineation for external and internal radiotherapy, response prediction, and monitoring. The molecular characterization of an individual tumor guides the choice for the radiotherapeutic strategy for specific patient. So it also becomes the road to personalized medicine because a different type of therapy could be planned for a different subject or the different human subject. And it is greatly elevated by the improved accuracy and safety as compared to biopsy with regard to discrimination of the heterogeneity of a primary tumor and between a primary tumor and metastasis. So whenever we do the biopsy, we take the primary site tumor, but we are not able to see what is happening inside the body, whether it is metastasizing. So metastasis stage would only be seen when we are using these radio diagnostic or radio tracer technique to do the diagnosis. And the same way, coming to the prostate-specific membrane antigen, means we can change the vector probe and we can design a system wherein we can label it with different radioisotope of interest, even for the radiotherapy or radiodiagnostics. And this could be utilized to kill a cancer cell. So this is one of the examples where you can do the simultaneous radio diagnosis and therapy. When I'm only using 125 iodine, which is a gamma emitter, I could only be doing the spectrometry. But in the same system, at one place, if I'll be using iodine 125 or astatin 211, I'll be doing the imaging and therapy at the same time. If I put the subject under the scanner, I'll be able to get the image. And at the same time, alpha therapy is being done with the same vector. So this is called simultaneously or radioisotope couple for tumor diagnosis and therapy. Now we have seen a single biomarker for a single receptor or enzyme for a single, how we can do the signal amplification using two-step targeting for diagnosis and therapy. So this becomes very, very useful when we are working with a long-lived radioisotopes. So we have to design a system wherein we can functionalize a molecule with a probe or with a macrocyclic agent, which is able to hold a long-lived radioisotope of interest and amplify the signal at the target site. So this has been done in two-step targeting, where avidin has a very high affinity towards the glycosylated proteins on the cancer cells. So first, the cold avidin or without labeling, the avidin is being injected, and then biotin labeled macrocyclic agent, which is complexed with the radioisotope or the radiometal for either for diagnosis or for therapy, is injected after a while. So advantages of these techniques are when you do the signal amplification means. So avidin binds to 4 units of biotin. So when the avidin is there in the body, after the clearance, its pharmacodynamics, we'll check the pharmacodynamics and pharmacokinetics. After the clearance of avidin, which takes place around in 6 hours, the biological half-life of avidin is around 6 to 8 hours. After 6 to 8 hours, biotin is injected, which shows 4 times signal amplification, or how much dose you can deliver is 4 times the dose you can deliver of the radiotherapeutic isotope and increase the killing of the tumor tissue at the target site. So now coming to these are all about the PET or the positron emission tomography. So labeling of these PET probes is really very cumbersome and it is challenging because we need to incorporate this radioisotope inside the molecule. So it requires like for making L-methionine, which is labeled with carbon 11, we need to take the precursor as L-homocysteine. And the ring is opened and the carbon is attacked with carbon 11 methyl iodide. So we get carbon 11 methionine, which is indifferentiated from the methionine itself, from the natural native ligand, methionine. So in terms of specific activity, it has got a very high specific activity. It has got a very high volumic activity of the radioisotope being incorporated into the molecule. But at the same time, it is strategically challenging to incorporate those short-lived radioisotope, non-metallic radioisotope into the biomarker, for which we have designed few labeling procedures by which we can design any of the biomarker of interest with carbon 11 and FAT. Now coming to the single photon emission committed tomography, the molecular design or the fundamental of designing this kind of radio pharmaceutical is quite different from what we have seen till now. So the basic principle remains the same. A radioactive labeled pharmaceutical is administered to the patient, and depending on the biodistribution properties of the radiopharmaceutical is taken up by the different organs or tissue types. There it decays, emit gamma rays. Then gamma rays travel out of the patient body and are detected by scintillation camera system, which is used as an imaging device, and so on. The resultant image gives the physiological state of the organ. So how it is being designed? If you see, this is a image of a folic acid, where we conjugated this to a macrocyclic agent or the macrocyclic system, which tags the folic acid receptor targeting strategy. It makes use of the folic acid as a molecular Trojan horse for selective delivery of the attached probes to FR expressing cancer cells. So we have limitless potential of this kind of constructs, because if you are using a macrocyclic chelating system, this same entity could be utilized for SPECT imaging. Same entity, if we use copper 64 or gallium 68, it could be utilized for PET imaging. If we are using gadolinium or European-based systems, that could be utilized for magnetic resonance imaging or optical imaging. At the same time, if this same construct, if we change the radiometal ion to rhenium 188, bismuth 210, astatine 211, it could be utilized for therapeutic. So in this way, this whole molecular entity was designed for SPECT-based system. And I've just told you the limitations of carbon-11 methionine. Carbon-11 has a very short half-life, only of 20 minutes. So it cannot be transported to any other hospital in per se. So to overcome this particular limit of carbon-11 methionine, we have come up with the design of a new biomarker, which is labeled with technetium, which is gamma photon, which emits gamma photon. It's a gamma emitter. It has a 6-hour half-life. So it can be transported to anywhere where SPECT camera is there. This molecule could be utilized to detect whatever methionine-based molecule does, like delineation of the tumor. It could be utilized for prognostication. It could be utilized for response therapy, non-responders, and seeing the responders to non-responders. So this has been indigenously developed, carbon-11 methionine, based on carbon-11 methionine. So here we have taken the two moieties of methionine, which was conjugated to DTPA molecule, which is the chelating system. Amino polycarboxylate system is introduced between the two methionine subunits, where it binds the metal ion. So technetium MDM diagnosed brain tumor and breast cancer with high precision. It is a cost-effective substitute for amino acid PET imaging. It is also used as a marker for evaluation of therapeutic efficacies and to differentiate between responders and non-responders. So for this, multicentric trials are going on. So very simply, we have designed this molecule and utilized this into more than 400 subjects or human subjects at our center and at PGIMR Chandigarh, AIIMS Rishikesh, AIIMS Delhi, and EMHANS recently. So coming to the MRI, which is magnetic resonance imaging. So what are the contrast agents could be utilized or to develop some of the contrast agents for getting MRI or seeing at a molecular level what we can do? So it is always that seeking a better contrast with molecular magnetic resonance imaging. Here, the contrast agents are widely used to increase the contrast difference between normal and abnormal tissue. So when the conventional MRI, we do the conventional MRI, we're not able to appreciate the pictures or the scans that we get, because in those scans, you're not able to differentiate between a proliferating tumor or radiation necrosis. So we need to develop some contrast agents so that contrast could be increased at the site. For example, in this particular scan, you can able to see that before contrast, we could not see the abnormality in the scan. But after gadolinium-based contrast agent was given, a high uptake in the meningioma or bright lesion could be seen in meningioma scan. Magnetic resonance imaging, it focuses on smaller and more selective areas than X-rays or CT scan. Three important parameters could be, determine the image viability, like water content, blood flow, relaxation time. So in this, MRI contrast agents are usually paramagnetic metal system, which functions by altering hydrogen nuclei of water protons and are classified as T1 or T2 agents. There are four types of magnetic fields. Diamagnetic agents are useful in gastrointestinal imaging. Superparamagnetic and ferromagnetic agents, which have net magnetic dipole larger than the sum of the unpaired. Dephasing of protons shortens the T2. Paramagnetic agents include iron, magnesium, gadolinium, dyspropium, all of which have unpaired electrons as a result, positive enhancement. So these are the MRI contrast agents. One is the DOTA. Sorry. MRI contrast agent, there's a DOTA ligand structure. This is labeled with, or complexation or coordination is done with gadolinium to reduce the toxicity of the gadolinium chloride. First, gadolinium chloride only used to, increase the contrast because it used to transfer the charge on the nearby proton. But the toxicity or nephrotoxicity was very much associated with the gadolinium chelates, gadolinium itself. The chelates were developed for entrapping the gadolinium, and then the charge transfer takes place from this gadolinium chelates to the nearby protons. But still, the toxicity levels are still high. It leads to nephrotoxicity, gadolinium-based contrast agents. So then the next generation came with the manganese contrast agents. Again, manganese contrast agents also have, similar toxicities with the magnesium. So again, there is dyspropium-based parashift contrast agent for develop that can reveal the temperature of the tissue. But there is a lot of scope in development of MRI contrast agent as such, because till date, only gadolinium-based, Magnavest or DOTA-RAM, these contrast agents are available for MRI contrast. But now the classification, if we go about the classification, we can maybe categorized according to the following features like magnetic properties, chemical composition, presence or absence of metal atoms, route of administration, and effect on the magnetic resonance image. So there are several, classified into T1 contrast agent, T1, T2, T2 star contrast agent, then CEST agent, F19 based agent. Excuse me. So then there is hyperpolarized imaging agents also that are there, which are based on F19. Heteronuclear, again, F19 based. So these contrast agents are still in infancy, and they are for the clinical utilization. This has to be worked on for the MRI application. So now, how you can these three modalities, how they're useful in optimizing the early drug development. It can also individualize the therapy or the response therapy by doing the patient stratification, early response monitoring, design of the rational combination therapy, and also monitor expression during treatment, optimization of treatment timing. So the treatment timing could be reduced if you are doing the diagnosis with PET and SPECT, and also seeing the coordinates where it is located through PET MRI hybrid scanning or SPECT CT hybrid scanning. So with this, I would like to take any questions if there are.
Dr. Amita?
Yeah. Dr. Pooja, thank you for such an elaborate and lot of new things I was able to learn also. So being in the pediatric oncology, my request will be to just speak about the theranostics in pediatrics, why we have not been able to translate whatever has been done in adult for children, because they need more precision medicine, more customized treatment.
I'll give you an example of our center only. Like if you've seen fluorinating dopamine scan that we do. So in 2013 or 14, we have got this one pediatric patient, which is was only 6 months old. So he wasn't when he gone into coma for some reason. So that was not very well established, and what all the biochemical testing was done and all the things were done, but they're not able to see that how this pediatric patient has gone into the coma. That patient came here. We have injected 1 millicurie, like very small amount of F-dopa in that patient or the child, and we could appreciate like in the basal ganglia, and we were able to see that the uptake is really nice in the basal ganglia. So this was sure that nothing is happening inside the basal ganglia, or the coma is for some there's some other reason for the coma and not the basal ganglia. So doctor were very they were thinking that the basal ganglia, if the basal ganglia is not working, that they will not be able to treat the patient in a nice way. So this has helped them to take care of the response therapy or the therapeutics for that patient. So in a similar manner, we have also done neuroendocrine tumor gallium-68 labeled dotatate in RNR hospital. It's in a 12-year-old boy, where you can see if I have the pictures, but not on this laptop. There's a huge accumulation in the abdomen region of the neuroendocrine tumor. It could, we have seen that in that case. Then it was subjected to chemotherapeutics, radiotherapeutics, and after that we have done the follow-up scan with the gallium-68 labeled dotatate. So it was very surprising that on the same boy, we got a negative scan after the therapy, and so no few millimeters also not detected on that dotatate scan. So he was completely cured of that neuroendocrine tumor, and then we also do the follow-up after 1 year, or it came out to be negative. So this neuroendocrine tumor, we have even given in 2013, we have given gallium labeling strategies to the hospitals. We have provided the for labeling dotatate to be utilized at the PET centers. So it is not that we are not using. Yes, it has not been utilized completely in the pediatric situation, because pediatricians, you're also a pediatrician. Radiation burden also there, you'll figure out that benefit to risk ratio also involves the radiation burden to the patient. So for that radiation burden, to minimize the radiation burden to the patient, we inject in very tracer amount. The activity is injected into very tracer amount, where we assess their benefit to risk ratio. So we have this all the molecules that are available that I have shown here. This is the study that we have done at our center. So maybe Dr. Maria will be taking the human subject, and you'll be she'll be taking up the follow-up and that response to treatment. So you'll be get to know that how these radio tracers are helpful in designing as the therapy for responders and non-responders. There's a very huge study that was done for the responders and non-responders in case of methionine, where we were able to assess that this therapy is not working for this patient. This therapy is working with this cohort of patients and not with this cohort of patients. So responders to non-responders also study we have done. Even we have hyphenated the technology by introducing MRS into the picture, where I have shown the picture of FET, which is done in Apollo Hospital. So what they have done is, with the uptake, they have also assessed the MRS. Magnetic resonance spectroscopy also they have taken into picture. And there's a paper that was published in 2015 on this from our center and from Apollo, that we have seen that the prognostication and how prognostication by this radio tracer could be helpful in designing the treatment response therapy and staging or grading of the cancer. So these radio tracers, all radio tracers could be utilized in pediatrics in a very well and assessed manner, assessing the benefit to risk ratio. So this could be done like for that.
Thank you.
Yeah.
Any participant?
Yeah, I myself want to ask one question from say Pooja, that why there are certain nanomedicines have been made out of the theranostic agents. So have any one of them has gone to the clinic or something?
Actually, the thing is that for radiopharmaceutical, we have a radiopharmaceutical committee or regulatory approval. It depends upon the biodosimetry of the radioisotope. But when you talk about the nanomedicine, you are labeling it with radioisotope. Now, nanomedicine itself has a different ethical committee. Because of this, like toxicity of nanomedicines are not yet proven. So there is there are papers which are there for the nanomedicine, like for the delivery in the brain. There are a lot of papers that are at the preclinical level, but it has not seen the light in the clinical scenario till date.
So like you were mentioning that there are clinical trials which are in progress.
Yes, ma'am.
Can you just elaborate a little bit that on what particular cancers those clinical trials are there?
Yes, ma'am. So I mean, initially that carbon-11 methionine we have developed for the proliferating tumors. So carbon-11 methionine we have already done for grading the high-grade gliomas and low-grade gliomas. Then it was, two projects were taken up, one for the breast cancer and another for the brain tumors. So breast cancer has given a very good, SUV and standard uptake value of this methionine-based compound, which was assessed, and it was made a tool to, diagnose or give the prognostication that this particular breast tumor will become malignant. That could also be defined from that project with the use of that methionine-based marker or the radio probe. Another study that we have conducted with the head or the brain tumors. Initially, we have screened a lot of patients with brain tumors, known brain tumors cases. So we have seen in the proliferating tumor for the assessment of astrocytoma, oligodendroglioma. So these kind of gliomas, they were very well, we were able to delineate, prognosticate, and also differentiate between responder to non-responders responders from the therapy. So this has become a very good tool. Means MRI could not give you any information on the recurrence. So this has also been used in recurrence of the tumors. So if a tumor is surgically removed, and then there is a recurrence, then also this methionine-based probe, very simple probe, very less molecular weight, it could be able to detect the recurrence also in the clinical scenario. This study was conducted at AIIMS Delhi, where they have seen the recurrence was very nicely seen with this proliferating probe that is methionine based. So this kind of different studies were taken up for different type of tumors, and it was assessed. So this I'll take one another, hour to explain all the things that are there, because histopathology was done, MRS was done. So on those biopsy lesions, the one which we have treated, the treatment was done, and monitoring was done with methionine. That also histopathology stages going from to a near normal, I would say. was also assessed by different techniques and corroborated with the methionine radio probe.
Okay, and then I would like to ask some background. So I would like to ask you one thing more, like, for example, if we are doing the cancer therapy using, we are doing the chemotherapy, radiation therapy, or the surgery. So what do you think? There are two parts of theranostic agents. Like, we want to say that it is being used for the diagnostics and it is also being used for the therapy. So what do you say that if we are using these agents like technetium based or gallium based, or you have talked about almost a lot of other agents. So if you are using DOTA or something, so will it be, should it be in future or maybe in another few years, do you think that it should be used as a from this as soon as the patient comes to you and the oncologist or the radiation oncologist or the physical or the clinical oncologist start designing a therapy, because they decide their radiological board sit there. So is it should be initiated, it should be used initially only, or in the later part of the therapy? Where should we use it?
Yes, Tom, it's a very, very nice question and very well taken that. First of all, theranostics, when we are talking this term for the radioisotope,
yeah,
we need to understand that theranostic, when we talk about radioisotope, it is sequential theranostic or it is simultaneous theranostic. Sequential means I have done the first diagnosis with a gamma emitter. And secondly, then I'm doing a sequential theranostic with alpha or beta emitter. That becomes a sequential theranostic on the same construct that we have made. Now, if we talk about the simultaneous theranostics, it's a very good example is samarium 153, where it is beta also and it is gamma also. So at the same time, with the same radioisotope in the same construct, we give it for the bone pain palliation and the metastatic lesion. At the same time, the therapeutics is happening, and at the same time, we can monitor for 30 days, we can monitor the patient under the scanner. Now, your question is, on which stage we should inject or we should use this radiopharmaceutical for our management or the treatment planning to be designed. So, ma'am, in this case, you know, FDG in India, FDG is available in all the PET centers, but not these specific targeting probes are not available everywhere. So right now, the procedure that the clinicians are following is they're heavily dependent on the FDG scan. They do the FDG scan at the time whenever the patient reaches. Nowadays, the scenario is this: the patient is reaching to the oncologist. Oncologist referred a PET scan or a SPECT scan according to availability. ये पहले ऐसा नहीं था. It was like that they should go through the biochemical parameters and all. Maybe Dr. Amitabh would share more light on this. That how, because he is into pediatrics, and if I talk about the normal, like the normal subjects or the cancer patients. So cancer patients that are being referred to us, like we have the case history that we have seen that they have gone to the doctor and they've come over here. So they are being, now they are being, like doctors are also, they want to see the PET scan first before they could go to any other observation or records. So they are also into the imaging modalities, all the imaging modalities they want to use. So the clinicians also, they are becoming very, very, they want to do the personalized medicine by using all these modalities. So the era has changed that now they are heavily dependent on these imaging modalities. Even like, for example, now if you go to an oncologist with an MRI scan, that there is some abnormality in the scan, the oncologist definitely will say that go and get a PET scan or SPECT scan done according to the availability. They will not assess any treatment planning on the basis of MRI. This is there. Like Now this is the things that are settled with the PET and SPECT scan. So once they are like, if I talk about FDG, which is really non-specific because it goes to infection, it goes to inflammation, it goes to any cancer where glycolysis is happening. So major hindrance comes when you talk about the treatment of a brain tumor. So FDG is avid in the whole brain. You're not able to detect a small fine lesion with FDG. So what is the solution? The solution for that is that the specific tracers that we have made based on methionine, based on choline, they are able to accumulate only at the target site. You are able to delineate the tumor. Delineate means you're not, whenever you're doing the surgery also, you're not going to miss any of the tissue that is cancerous during the surgery. First thing. Second thing, I want to quote one example, very nice example, because we have a lot of surgeries we have stopped due to this molecular imaging scans. So one scan we have done with the fluoroethoxytyrosine, or because already we were doing at Apollo Hospital. So this is the case of the Gangaram, where they have they have sent few images of FDG avid. Very big lesion in the brain, and they were saying that we need some modality to see that this is tumor or this is something else, because the biopsy, they were not conducted any biopsy on that. On the basis of FDG, on the basis of standard uptake value in that patient, they have planned a surgery of that patient where they have to cut out the skull of that person to take out the tumor. So this was all planned with the neurosurgeons. So this radiation oncologist and neurosurgeon, or the neuro-oncologist, they have this issue that neurosurgeons or the neuro-oncologists, they were saying that we have to operate immediately. And this radiation oncologist, they were saying, no, you have to wait, because we need to assess, we need to see that it's really the cancerous tissue or not. So radiation oncologist, they had a doubt on that FDG scan that it could be something else. But neurosurgeon, they were like, so they've asked us that, can you provide something which can differentiate between benign and malignant tumor, or maybe some. So they were not knowing that we were producing fluoroethoxytyrosine. Then with this, tyrosine was labeled and sent at that hospital. They've injected 3 patients. One is the positive patient, known positive glioma patient, they've injected. One, they have injected a known negative patient, or the healthy volunteer they've injected. And third, they've injected on which the person on which the neurosurgery was planned to take out the tumor. To their surprise, the neuro-oncologist surprise, not the radiation oncologist. So they did not find any uptake of fluoroethoxytyrosine. So the neuro-oncologist who were planning for the surgery was now, they had a management meeting wherein we were also like the part of that meeting. Then now FET is not going, and then the FDG is super avid and it has a very high standard uptake value. What to do with this case? Whether to operate or whether to not to operate. So in that case scenario, the doctors, they went on to taking a biopsy sample from the nose, doing the microsurgery, and they have tested it for histopathology. It came out to be a fungal infection. So just imagine, this is the power of molecular imaging. Like we have stopped several surgeries in the same manner. This is the one case I'm talking about, but there are several other things, several other surgeries that has been stopped with this by the use of this molecular imaging. Now the hyphenated imaging is also possible. We are also assessing with the MRS, with the PET, with the receptor quantification. The image that I have shown to you for the Parkinson, where you could see a very low uptake in the basal ganglia. There is an image where I have seen the Abdopa scan where the basal ganglia were The uptake were very nice in the normal healthy volunteer, and that is a very we can do the receptor quantification. Like if the normal receptor D2 receptor is this much, and it is coming out to be this much, this imaging modality has a lot to do with the psychiatry also. When you psychiatric disorders, when we talk about the psychiatric disorders and we talk about F-dopa or we talk about Daclopride or postsynaptic and presynaptic receptor imaging, this particular radio like probe would be useful in determining the receptor quantification. You have heard about cognitive overload and you have heard about cognitive fatigue. These two things are different. But when you see the symptomatic symptoms in the patient, they are very similar. In case of depression, also you will find a similar pattern. There are different battery of tests that neurological testing was done. Neurodeficit scoring is done, but the symptoms are same. But when you assess on the F-dopa scale of the F-dopa scan, you will find out that how much receptors, like I am treating 2 patients in psychiatry, both they are suffering from depression, and I'm giving on the basis of symptoms, on the basis of what symptomatic they are showing, I'm writing the same medicine, levodopa, to both the patients. One comes with the tremors, and one comes with the good biological response. It is showing the responding to the therapy, and one is not showing to the therapy. So this neuroimaging or the neurocognitive disorders, for this also radio imaging probes have become a very essential point in treatment planning, because 3 milligram of levodopa is not sufficient for another patient. So in psychiatry, if you talk about receptor quantification becomes very, very important, because you're dealing with very, very low concentration even of the therapeutic entity like levodopa or any other antidepressant drugs you talk about. So in that case scenario also, this has proven to be a very good tool in addiction, in schizophrenia, in depression, and in cognitive overload and fatigue. So nowadays, psychiatrists also looking for the neuroimaging with the radio tracer, and only after that, after assessing the radiological imaging, they are doing the treatment planning.
Okay, great. There still I have a couple of questions, but I think if you are staying with us in the next session for maybe another half an hour, it will be great.
Yes, ma'am, I'm there.
So great. So if you stay there, it's fine. Because we have to move to the next session. It is tutorial, and I have kept your name there.
Yes,
ma'am. So although we are talking about the clonogenicity and that, but I would like to continue with you for some time for this also. So is that Dr. Kale or Dr. Amitabh want to give some comments?
I will request Amitabh only, because he was following very nicely.
Yeah.
Okay.
And madam, I hope you will permit me to leave after some time.
Yes, I know. That is why I stopped puja, so that because you are
No,
from the morning.
Okay. No, it was very interesting, and I can postpone a little bit, 10-15 minutes, no problem.
Yeah. So yeah, I'll leave after that. And Amitabh, do you have anything to comment on that?
No, I have
oncological point of view.
I have also learned a lot from Dr. Puja today. And these are exciting things to hear, because in oncology, you need to know that we are moving in the right direction. And hope is something which we all give to the patient, but the doctors also need to, okay, new things are on the horizon, and it will become easier and easier for you to take treatment decision and all. So thank you, Dr. Puja, for such excellent talk, and such nice examples also you have given. So that's all from my side, ma'am.
Okay, so I would like to thank, Dr. Kale and Dr. Amitabh, but although Amitabh, if you have time, you can stay with us. Dr. Kale is with us from the morning, and there was another chair who was not present, so I just requested him, and he very kindly agreed to be with us. So I would, before concluding this session, from every chairperson, I've been asking that that what is your own area. So I would like to hear from Amitabh, so that our students and should know that in what particular area you're working. And I know that clinicians have very small time to do the research, but still, whatever time you have, maybe you can be benefited by the others who are present here. There are a couple of faculty here from Gangaram, AIMS, and other our molecular medicine faculties are there, some SLS faculties are there. So they may be also benefited, and maybe you may also think about it for the collaboration, because one of the major objective of the GIAN is the institutional collaboration and international collaboration.
Yes. Thank you, ma'am, again for the opportunity and inviting me. We have a large pediatric department, one of the largest government sector pediatric department in Delhi NCR. And I am part of that department, and the division is Pediatric Hematology Oncology Division. And we are basically dealing with pediatric malignancies, benign hematological disorders like thalassemia, ITP, and also inherited immunological disorders. So the main cancers we are dealing with is acute lymphoblastic leukemia, myeloid leukemia, NHL, HL, and beside rare tumors also from time to time. But these are the four main malignancies which we are dealing with. And my present area of research, where some research is ongoing or has been completed, is risk stratification to define, to find out the right treatment, dose, less intensive treatment for children who don't require a high dose chemotherapy. Also the biology of pediatric cancers, and social and nutritional aspect of pediatric cancer, because our client is, is the poorest of poor. So there are a lot of other aspects which affects the outcome of cancer treatment. And also drug monitoring and predictors of outcome, as I have already told. And infection in neutropenic patients also is one of the core area of our emphasis. And late effects of treatment of cancer for children who have completed cancer treatment. These are the current area of research we are involved in, and we'll be happy to collaborate. Thank you.
Okay, so fine. It's nice to hear from you. And now I would say thanks to Dr. Kale for being with us. And just for the information for the Amitabh and Dr. Pooja, Professor Kale is a radiation biologist, I should say, or not. He was a chemist actually, and he spent a long time in School of Life Sciences. He was a vice chancellor, and I would say that he is one of the pioneer in the area of the radiation. When people don't used to believe in the radiation biology and radiation oncology, at that time he taught in School of Life Sciences. So he has like I would say that created a whole progeny, I would say, that whole number of radiation biologists in the India, and some of the faculties are here. And so we can always benefit from him, and he has worked largely on the, say, plant materials. And there were, so I would say that lot of students can also, now he's superannuated and he's settled in Pune, and but students can always contact him, because you can learn. There is a problem today which we were discussing from very beginning of this school, that there are very few radiation biologists are there, or we are not teaching radiation biology anywhere. I only came to know yesterday that Mata Vaishno Devi University is giving a certificate course on radiation biology. So so people like Professor Kale can always be help students in working in that area, or creating an understanding. And I will certainly invite you, sir, for the next time for the giving you lecture, so that our students should be benefited out of it. So thank you, sir. If you want to You want to say something, you can
No,
say.
no, Mata, what I can say after this one, I'm overwhelmed by your comments and observations. Great. I'm really grateful to you. I don't think I deserve that much compliment. But anyway, thank you very much and giving me opportunity.
Thank you.
So, yeah, I will. I'll be for some time, and then I will, in between I will leave.
Okay, fine. So the next session, because Pragya is not there, so she has. So I will start the next session, and next session we have put it like this, that we are having the tutorials in each session, and in the each and every session we are. So we have talked over here that we will be giving some demonstration of the growth kinetics, cytogenetic damage, and chromosomal aberrations. So we can do one thing, that we can, for this demonstration, for the students' benefit, we have created some videos. So we have done a say 20 to 27 minutes video there. And so I think I should, whether I should hold Pooja and tell her to come back, or we can, or I was just thinking, Pooja, that if you have time, or if you have certain images somewhere where you can talk something in terms of the tutorial, and you can make some something, and by the time I talk to them about this growth kinetics, and then after 20 minutes, can we resume with you? Because I think the kind of information you are going to give, we don't have any other speaker of your say your specific modality. So, or would you like to show some of the discuss more here only now, or after 20 minutes? What would you like to do? See, I have a video and about the growth kinetics. So we will display that video, and after that I will tell them that, so they will tell you how to do, they will growth kinetics, cytogenetic damage, and then I will tell them that what is it. So because I wanted to show them, because we have some very young students also there, because if they don't know what is growth kinetics, how would they really understand it? So we have made videos. So tell me, how should we go? Should we finish?
Yes, ma'am. Maybe yes, ma'am. I am open to both. Like I can join after 20 minutes, or I can do it right now.
Okay. So you have some of the, you want to do it now?
Maybe
I think that will,
what I can do is like,
I think that will be better actually for the continuation. So people will be listening that one and have connect with earlier continuous lecture. It is up to her. It is up to her.
Yeah, Pooja.
I'll be requiring 5 minutes to transfer my slides from this desktop.
Okay, so let me talk to them, and then by the time I'll talk to them about the growth kinetics, maybe let it be for 20 minutes, and then you can come back. Okay. Sure. You are here only. Dr. Amitabh, if you want to leave, you can leave.
Thank you, ma'am. Thank you.
Yeah. Thanks a lot for coming. Thanks a lot. And you can join on the other days also.
Yeah, sure. I'll do that.
Yeah, we have a full session on imaging. Again, by the Pooja's colleague only.
That will be more oriented towards the human subject and the outcomes with the patients.
I'll definitely join.
The clinical lecture.
Yeah, that is on 31st. That's completely on the human imaging. Yeah.
Okay. Thank you.
Yeah. So now I'm talking to the other students. So other participants, that if you really there are whenever you try to see the growth kinetics, you are trying to see the effect of the radiation. What you do, that you have to do certain experiments. It is not only radiation. If you want to see the effect of any drug, any small molecule, any kind of say any kind of a say therapeutic agent which you are using, you have to find out, you have to do certain pharmacological testing, certain cellular biology assays, cell biology assays. So for doing such assays, there are certain, so in the the radiation, as from last 3 days you are listening, there are some gold standard assays like clonogenic assay, growth kinetics. Growth kinetics is done even in the presence of the drug also. And in the presence of any other material, you should do the growth kinetic assay. And then clonogenicity assay. So we have, so how you have to do it in the in vitro conditions. So we have prepared a video. And in general, I would like to inform you that every cell, whether it is inside your body or it is in a plate, what you do that it has a certain growth kinetics. It has a doubling time. It has it doubles in 20 mammalian cells. Generally, people say that it doubles in 24 hours, but that is not the truth. There are certain doubling agents which actually double more than say after a month or 10 days or 15. So it depends on the cell type, which particular cell type you are using, and that depends on the doubling time of that particular material. Is cells are dependent on the cell type. So depending on so what we do that in order to see what kind of effect is occurring on that particular cell in the presence of a small molecule, or in the presence of radiation, or in the presence of any kind of a stress or inflammation, we try to see the most basic the assay which we do in the laboratory. That is, we try to see the growth kinetics. Even in the case of the bacteria also, we see that what is happening with the bacterial growth kinetics in the kind of some stress, or in kind of a certain say exposure, or some kind of infection when it is in human, when it is causing infection in an organism. So there are different kind of say methods are available, but the growth kinetics is supposed to be one of the most basic say experiment. And I would like to run a video in front of you, which is actually performed by you, by the Antra, who is the PhD students, 4th year PhD students in my laboratory. And she will try to tell you, and after that 27 minutes of the video, we may discuss it, but I would say all of you to hear it, and it may be very fast for you. Then you can discuss with me if you have any question, and Antra is also here to answer your question. So I would like to just play it for you. So
hello everyone, I'm Antra, a PhD student working under the guidance of Professor Vidha Tandon, and today I'm going to demonstrate cell growth kinetic assay. This is a commonly practiced experiment in the cell culture technique where we check the growth of a cell line, the doubling time through the growth kinetic. So today I'm going to present the growth kinetic of HeLa cells with treatment of a drug and the radiation. So HeLa is So I have seeded the cells in a 90 dish and kept in a CO2 incubator, and debris and the cellular particle released from the cell. We give a wash with 5 ml of TDS. Then we go for trypsinization. So we take 0.5% of trypsin EDTA solution, and for a 90 dish, we need 1 ml of trypsin, as we set the pipette to 1 ml, and we take 1 ml of volume and spread it over the surface of plate and spread it properly. Then we have to keep the plate under the CO2 incubator for 2 to 3 minutes, so the cells will get detached from the surface of the plate. So after giving trypsin treatment, we keep the plate in the incubator for 2 to 3 minutes for the cell to get detached. So after 3 minutes of incubation, we take out the plate. We can check the morphology of the cell under a microscope. Initially, the cell morphology is elongated. They are in dendritic nature, but once you trypsinize, the cells become spheroidal. So you can check in microscope, and we are going directly for media addition. So here, to activate the effect of trypsin, we will add 2 ml of media to the plate. Now we'll make cell suspension. Using the pipette, we will mix the cell and dissociate them into single cell suspension. Then we harvest the cells in a 15 ml falcon. We take 2 falcons, one for balance and one for the cell suspension. So first of all, we'll add 10 ml of media. For the balance, we can use PBS. Add the cell suspension into the media. To balance the volume, we have to add more PBS. Then we'll centrifuge the falcons to pellet down the cell suspension. So we move on to the next step. So this is our tabletop centrifuge. We turn on the instrument. We will keep our falcons in a balance. Close the lid and we set the speed of rotation 900 RPM and time 3 minutes, and temperature should be 4 degrees. We set the temperature at 4 degrees. Start the run. After 3 minutes, the cell will pellet down as a big pellet, and we will use it for feeding our plates. Now we can see the time is over. So now we open the cover, take off our falcon, and you can see there is a big pellet, and this is the HeLa cell pellet. So now we'll wash off the media and proceed for the feeding. So we turn off this instrument. So once we have centrifuged the cells, we'll discard the media. We suspend the pellet in 1 ml of fresh media. We suspend the pellet using pipetting. Now we'll go for cell counting. So from the solution, we will take 20 microliter of cell suspension and dilute it with 80 microliter of Triton blue to get a dilution of 5 times. So we take a 1 ml Dippin' box. Here we have 2 ml. We use this. So this is Triton blue dye, and we will use the method of Triton blue exclusion method, where the viable cells will exclude out the Triton blue dye, while the dead cells will retain the dye, and we will see the blue color cells which are dead cells. So we will take 80 microliter of Triton blue. Using a 200 liter pipette, we will take 80 microliter of Triton blue. Before taking the cell, we have to resuspend it again because the cells can pellet down. So we mix the cell first. Using a T2 pipette, we will T20 pipette, sorry. We will take 20 microliter of cell suspension and mix it with the pipette. Mix it properly. And then using a cell counting slide, and this is the automated cell counting slide that is used for automated chamber. So we will take out the slide. So as you can see, we have two loading platforms here. So we can load from the left or right side. So here we are loading from this end. We can load up to 10 microliter of our sample. So we set the pipette to 10. And load our samples. 10 microliter of sample. Check the number of viable cells in a countless chamber. And this is an in vitro instrument where we can count the number of cells directly. So over the side where we have loaded the sample, we will insert it here. And once you insert it, you have an option of count cells. So we go for count cells. And it has started counting the cells. It will take 20 to 30 seconds and then we'll get the number of cells. So we wait here. So the number represented here is 1.6 into 10 raised to power 6 cells per ml. So this is the number of cells we have per ml. So now we have to seed the cells in our plate. So we proceed for the next step. Here, because we have 4 different groups, so I have labeled the plates already. We are going to use 35 ml Petri dishes, this one.
Why we are not able to
2 ml of volume. So the first group is control group where we are going to check the number of cells at different time points at 0 hours, 24 hours, 48 hours, and 72 hours. The second group is the drug-treated group. Here we are going to take the fluoroparazine drug. We are going to check the effect of this drug over the cell growth kinetics. So we will take the drug at a concentration of 200 micromolar. So these 4 plates are for the drug treated cells. And then we have a radiation group also where we are going to give 4 gray of radiation at 4 different and measure the cell count at 4 different time points. The last group is drug plus radiation where we are also checking at these same time points 0, 24, 48 and 72 hours. So we have to feed the cells for our experiment. So in each plate we have to take 2 ml of media, and for HeLa cells because they culture in DMEM, So we will take BMEM media, open the cover of plate. So in total we have 16 plates. So using a 10 ml serological pipette, we will add the media. 2 ml each. At the seeding step, we have to seed equal number of cells in all the plates. So this is our cell suspension, and we have a count of 1.6 into 10 raised to power 6 cells per ml. So we have to seed 0.5 million of cells in each plate. So we will add the volume accordingly. So here we are going to add 50 microliter of plate. Before adding, mix the cell suspension and add the cell. Once we are done with heating the cells, close the cover of each plate and mix the cells. Spread the cells homogeneously over the plate. We have to mix the cells by rotating the plate like a A shape and keep the plate in. Now we keep the plate in the CO2 incubator until it is adhered to the plate for overnight. After overnight incubation of our cells, we have to give the treatment of drug to our respective plates. So we have 4 plates where we are going to give drug treatment. The drug is chlorothalazine, and we are going to give 200 micromolar final concentration to the cell. Before adding the drug, mix the solution properly. Then we give the treatment of drug. Like this way we add to all the plates. Mix it properly and then we keep it back to the incubator and we will do this treatment for 4 hours and then we'll move for the cell counting. For the drug treated groups and for the controlled group, we go for the cell counting directly. And this is the 0-hour time point. So we count the cells like I have shown previously using the automated cell counting slide. And for the other group where we are going to give radiation, we will proceed to the X-ray irradiator machine, and which is situated in GIAN J2. We take the plate there by wrapping it with a paraffin and then irradiate. So once our cells get adhered to the plate, we go for the treatment. Drug treatment we have previously given, so now we are going for the radiation treatment. Here we have two groups, only radiation and drug plus radiation. So we are going to keep our plates inside this instrument. This is X-ray on, where we use X-ray irradiation, and we are going to give 4-gray radiation. So this is the stage where we keep our plates, and this is collimeter through which we set the field of radiation, and this is the distance SSD, which is set to 50 centimeter, and we can adjust it if the sample is different. If for in case we have animals, so the centimeter goes down, so we have to adjust it. But for plate, this is fixed. Then on the screen, we see that there is recall program. So we select the program and we go to the second option where the SSD is 50 and the filter is first and the dose radiation is variable. So we choose the dose of radiation. And here we set the dose to 400 centigrade, which is equivalent to 4 gray. So now we go to continue and start the X-ray source. Now this X-ray is blinking, so means the radiation source has started. And here the dose of radiation is like started. So once it achieves 400 centigrade, the radiation source will switch off. And this is the dose of radiation that is fixed. And here it is 140 centigrade per minute. So, and this is the screen where we can see the plates which are getting irradiated. So once the total dose is achieved, we stop the X-ray source. And now we can see on the screen X-ray is off, so we can open the door and take out our plates. And next we proceed for cell counting for the growth kinetic assay. So after irradiating the plates with 4 gray of radiation, we will keep it back into the incubator. And we will take the cell counting reading at 24 hours of time point. So similar way, we will count the cells for other plates also. So after 24 hours, I am opening the plate. And then we will trypsinize the cells and count the cells using the same instrument, the automated cell counting chamber. So like I have shown it earlier also. So based on the number of cells, we will plot a graph, that is growth kinetic assay plot. And we can plot the graph using Excel or using the graph pad. So I will show you the how to make the plot in the next step. So we have taken the readings of cells at different time points for all the 4 groups. So the first group is control, where we didn't give any treatment of drug or radiation. The second group is radiation, where we have given 4 gray of radiation. The third group is the drug treated, where fluoroparazine at a concentration of 200 micromolar is given to the cell. And the last group is drug plus radiation. So we are expecting that after giving treatment of drug and drug plus radiation, the viability of cells goes down and the doubling time is increased. So we have feeded the number of cells at all the 4 time points, 0 hour, 24 hour, 48 hour, and 72 hour. And we have gone to the analysis where we have taken the 9 dotted plot, click OK, and we generate this graph, and we can select the color coding. So I have generated a figure where we have given different color codes to all the groups. So this green color is showing the control group, where it is showing exponential growth at different time points after 24 hours, 48 hours, 72 hours. The cell is continuously growing, so the doubling time is not affected. But after giving radiation treatment, the cell growth has delayed and the cell proliferation time has increased. But if you give the treatment of drug, that is fluoroparazine, it is inhibiting the cell proliferation. So the proliferation time is increased. But if you give drug and radiation together, you can see there is a drastic change in the kinetics of cells. And if you compare it with the control, the cell growth has prolonged. So the cell is dividing very slow. And initially, because we seeded 0.5 million of cells, now we can see that the cell number is almost same even after 72 hours of treatment. So this is showing that radiation plus drug is affecting the proliferation of a cell line. So this is the use of the cell growth kinetic assay, where you can check the doubling time after treatment of any drug, radiation, or any other chemical treatment. So you can check the effect of these treatment types over the cell kinetics. Thank you.
So is there any question by any participants, or in between, if you lost the video, we will upload it on the YouTube GIAN website, so you can look at there. But if anybody has any question, can ask us. Hello? Hello? Okay, I don't think anybody has any question. So yeah, so Dr. Pooja is already there. I don't know today. Yeah, so Pooja, they may be would you like to share some your screen, or how would you like to go?
Yes, ma'am, I'll share my screen.
Okay. Okay. It's asking you join a meeting. Okay. तुम तो ऑलरेडी मीटिंग में, इसको बंद कर दो मेरे ख्याल से. Pooja, Dr. Pooja. Palak, just make her, she was out and she again joined.
Yes, ma'am. Actually, you said leave the meeting. I think that is also connected in background. So.
अच्छा अच्छा. No, I didn't say leave the meeting. मैंने कहा तुम तो already हो उसको बंद कर दो. अच्छा अच्छा. हाँ ठीक है ठीक.
So. My slides are visible?
Yeah, sure.
Yeah. So actually, we have learned about the imaging. It's the most powerful tool, I would say, in the imaging sciences that we have learned. So what are the other advantages or other advantages and advances in these modalities, which can be utilized in various other domains? Like it can be used as a tool for refinement and reduction of the use of animals to study model of disease. I thought this is a very important topic because whenever we are, conducting an experiment and doing some experiment, we are taking a lot of animals. Can we go away with the use of animals or minimize the use of animals by using teaching techniques? Is it possible to do that? So first question is this, and second is that can these modalities, the imaging tools that we look into, PET, SPECT, and MRI, can they help in the drug development as such? So these are the two questions. These are the two questions that could be answered by using or seeing the recent advances and challenges in preclinical imaging and how it can be used as a tool for refinement and reduction of the use of animals. So non-invasive imaging may play an important role in efficiently characterizing small animal pathologies. Whatever experimental model you develop, that can be assessed through these non-invasive imaging modalities. And these technologies scale favorably in providing more physiological information. It could also contribute to increasing our understanding to measure the indicative markers in disease state. So if you see that there are all preclinical imaging pictures and how PET, SPECT, MRI, CT, optical imaging and ultrasound imaging has, contributed in assessing the disease site. So a clear picture, if you see that PET can give you the molecular imaging, like molecular process pictures and not the anatomical details in the animal model. Now, if you see SPECT, if you combine with CT or if you combine with MRI, you will be able to appreciate that where these lesions are located or the radio tracer uptake or accumulation which is there in the animal, you are able to locate it. So to assess your experimental model, to characterize and to validate your experimental model, this becomes a very useful tool. So how it is going to be an improved tool? So better imaging means fewer animals or improved data. Yes, ma'am?
नहीं मुझे लगा तुम्हारी आवाज है. Yeah, it's fine. Go ahead.
So better imaging means fewer animals, improved data. Is it really so? It is statistically significant, or statistically it is acceptable to the whatever research work that we do. Is it acceptable if we do this reduction in the use of animals by using imaging? Or we can do the refinement, which can have a better handling and better pain relief, shorter experiment times, better housing. That can lead to, like imaging can lead to this refinement also. So how we can design a molecular imaging study by this? So first of all, we need to have a biochemical or process or pathological site. Secondly, we need to assess or see the molecular target that we are interested in. We need to choose upon the imaging modalities. Either we have to choose upon the optical, PET, SPECT, MRI, Raman, US, ultrasound. So these are the imaging modalities that are available. And are these modalities like it is directly significant to my research area or my experimental model? According to that, if I'm growing a tumor, how I can assess non-invasively, because every time I need to see and check, I have to invasively cut down that animal, take the measurement of that tumor that this much has grown, the tumor has grown this much. So what imaging modality I can use to assess that non-invasively, where I can also reducing the number of the killing, because I should know that what is the area or what is the volume of the tumor, which is inside or deep-seated inside the animal body. So in that case scenario, I will choose CT as my imaging modality to see and to assess or to monitor the increase in the tumor size or tumor volume. So CT would tell me that yes, whatever I have inoculated, the number of cells that I've inoculated has led to the tumor of this size at this. So you don't have to kill the animal as such. So non-invasively, you can see that your tumor is growing at after inoculating the tumor cells. So after that, if you want to assess your drug, like you need to do some tumor regression analysis, or you want to do some studies that you want to see that my drug is responding to that tumor or not. Again, what you have to do is you will go, start when the tumor is palpable, right? Then you will start your dosing. So why I'm using this, that the tumor is palpable and then I'm starting the dosing. But if you use the CT with your dose, when you are doing the dosing, what you can assess that when the tumor size was this much, I have injected my dose to the mice model. And then from that, you can assess the tumor regression in a quantitative way. You can see in the real time that what was the tumor volume and how you can assess that. So this becomes a very good tool for drug development. And then you can see that small animal ultrasound is also very, very important when you are talking about the molecular imaging using ultrasound in animals. It is relatively inexpensive. It will give you quantitative data, and it does not involve any non-ionizing radiation. It has good temporal resolution. But the key limitation in this is that it is not really deep-seated. It is limited to imaging soft tissues only, and no bone or air structures could be, seen on ultrasound imaging. And then you come to the fluorescence molecular imaging. Fluorescence molecular imaging also is a very, it has multiplexing capabilities. You can inject a number of markers at the same time and assess at the different wavelength. You can see in the real time. But the problem with this modality is that surface weighted image you can see very nicely, but deep seated, if you want to see into some kidneys or lungs or pancreas, so you are not able to see those organs with this modality. You have to do the studies which involve ex vivo experimentation. So here CT, animal CT, computed tomography, this has a limitless depth of penetration. You can go anywhere when you are doing the 3D CT computed tomography. It has good temporal resolution. You are able to detect the mass. Maybe you are not able to differentiate between a benign and malignant mass, but you are able to detect that where is that large amount of mass, or which is the abnormality could be seen on the CT imaging. But as you're developing an experimental model, you know that you are injecting a sensitive or the tumoral tissue or tumoral cells. So you know that this will cause malignancy. So this idea you are already having. So to assess or the tumor monitoring in your tumor regression studies, CT scan becomes very, very useful in drug development. And then small imaging MRI. MRI also is a very powerful tool because it has got very limitless depth of penetration. It has got high spatial resolution. And again, no ionizing radiation is there, but it has a poor sensitivity. Jab aap contrast agent, you talk about the contrast agent for MRI, you need to inject in milligram quantity. So like that already I have shown in my previous lecture, the gadolinium based compounds, they become little toxic also when you inject them into the patients. So, but conventional MRI could also be seen as a powerful tool to assess and analyze various other disease conditions pertaining to your experimental studies. Then come to the PET. Its key strengths are it has got excellent sensitivity. You will get quantitative data that where your radio tracer is going and accumulating in the brain. Even the small graphene nuclei, coded putamen, striatum, you are able to do the receptor quantification if you're using an animal imager for positron emission tomography. And the same way in the SPECT also at the molecular level, you'll be able to appreciate the accumulation of the radio tracer that you have developed. Even the normal probes that you're using for therapeutic application, or say only you're developing therapeutics, that also that therapeutics, that agent or that can also be labeled with some SPECT radioisotope to be seen inside the SPECT scanner. So this will help you in assessing your PK/PD profile without even killing the animal. It goes same for the PET and SPECT agents. Like you inject some imaging agent labeled with some radioisotope gamma, which emits gamma photon. Then you can assess the biodistribution in the whole body. At the same time, you can see the accumulation at the site. Also, you can do the quantitative analysis by calculating the dose at different organs. So you'll get the dose in kilobecquerel per cc in every organ. So you can also do the biodistribution non-invasively in the animal model. And you can see here, you can how much you can minimize the use of animal at this stage. And also you can reuse these animals because after some time the radioactivity will decay and then the clearance will take place from the animal body, and you can always reuse the control animals and your experimental models for some other study. So understanding the preclinical PET/SPECT CT imaging system. So what it has the system that we are having is a triple modality system in which we have the PET gantry where we do the PET imaging, and then we have a SPECT CT system in which SPECT and CT is being done, and we can co-register PET with CT and SPECT with CT. So these two are possible with this system, that we can also do the hybrid imaging in both the modalities. So here you can see the photograph of the animal bed with the mouse as a subject. Can be loaded and prepared while the bed is positioned at the front of the gantry. So when it goes inside the gantry, the injection can be done on the bed. In the real time, you can see the dynamic imaging. For 1 hour, you can see in the real time where your drug is going inside the animal and how it's excreting. So PK/PD could also be done through imaging modality. So this is the triple modality system at INMAS. So where we do the animal imaging, we have done the rat brain imaging also, and then this is the tumor. You can see that we have inoculated a UAD7 xenograft, and then we have this injected integrin-based radiopharmaceutical, and then after 1 hour we have taken the image, and you can see a very good uptake here at the tumor site. This is the blocking study that we have done with the peptide-based pharmaceutical, which was labeled with gallium. And then you can say that receptor blocking studies, when we have blocked this with the peptide, the 99% blockade was seen during the imaging. So here you can see that the tracer kinetics also you can study very nicely in this imaging modality. You can see that when my drug is accumulating at the tumor site, how it is rapidly clearing from the background, what was the counts in the blood, kidneys, liver, spleen, bladder, brain. So all these things can be done. The tracer kinetics could be calculated and very well calculated from this softwares which are available with this imaging tools. So these are the different preclinical imaging to study the model of disease that we have studied at our center. Musculoskeletal, we have seen some bone metastasis models that we have made. You're able to see that lung fibrosis. Was extensively been studied. The one group in our center has created a bleomycin-based lung model, and then the lung fibrosis was the studies was conducted, and we have seen that very nice results we have obtained without killing the animals. Though it was corroborated after that with the lung tissue histopathology. Now, if you see the CT scan of thrombus in mice lung, you can spot the difference between, this is the CT. And when we have created a thrombi model or thrombus thrombi model, we could appreciate that there is a CT can detect various clots in the lungs. So this becomes a powerful tool again to image these kind of experimental models. Also, we have done some neurodegenerative disorders experimentation where we have done the rat brain imaging. We can do the volumetric quantification also using the different software that are present in the preclinical system. Now, there one dental implant was done in the rat model. There you can see that we can definitely see the bone growth over the implant using these modalities. So ability of CT PET fusion to assist in visual anatomical localization of radionuclide uptake. So here you can see that both kind of imaging like PET-based, SPECT-based and effluorinatin-based imaging is possible in a single animal. So this is the amifostin analog DID30, Tanner's bleomycin-induced pulmonary fibrosis in mice that we have monitored through the CT that I have shown in the previous slide. Now, how to target these molecules like in the brain? It becomes very, very difficult to see and to make the molecules or the drugs for the brain. So what it requires, it has to cross the blood-brain barrier. When we are saying that it crosses the blood-brain barrier in real time, if we are developing some drug, we are not able to see that it is crossing the blood-brain barrier or not. So it becomes again imaging modalities. These becomes very important tool when we are specific drug candidate to be evaluated for neurodegenerative disorders application. We do the target validation. This all is done in vivo, non-invasively in animal model. So here I wanted to show you how we it's a movie, it's not working. So I want to show you that how we do the direct brain deformation and how we assess it in the CT. See, we will do the craniotomy, and when we do the craniotomy, you can see we remove the skull and this skull is like 4.23 mm skull we have take out. We take out this much of the skull, and then we do the impact on the open head. Open head injuries is given on this, and we assess this on the animal system, the surgery. So this is how helpful it is. So after that, MRI also becomes an important tool, because after impact, we need to see that the impact has, what damage it has created. That could be assessed through MRI. So this is the one of the DTI that we have done on the same model to see that hematoma is not formed. So we have done the angiogenesis sequence. We have run the angiogenesis sequence, and we have seen that there is no hematoma and the damage is in the serotonergic system. So these are all is possible because of the stereotaxis guarded animal models that we have prepared and then assessed on the and different subjected to different imaging modalities. Yes, ma'am. Yes, ma'am. I think that we all. Are there any questions?
I'm speaking and I'm muted. So yeah, so I have a like there are questions, but first I will ask certain thing from you. I don't know where I have disappeared. Can you see me?
Yes, ma'am. Now I can see you, and yes, ma'am.
तो कुछ मैं तुमको नहीं देख पा रही, मैं ये नहीं समझ पा रही कहाँ गए. Fine. So like when you are saying that you started from there, that how do you plan to do the imaging? How we need to do the planning, that how we have to do different kind of imagings. So I have a question here, that you showed it in the animal and you have said very nicely that if we do it in animal model, then by imaging it will be say non-invasive imaging, and the every time you don't need to sacrifice the animal and all this thing. But as far as I understand, so when we inject these animals with certain these theranostic agents, so so is there any kind of a toxicity is not there? Or you only because when you look at the gamma camera, or you look at by the different cameras, depending on the kind of radioactivity you're using, so is that there is no like somebody has asked the question, I'll ask it, but is there the toxicity is not a point over there? First I'm talking about the lab research, then I'll go to the human thing.
Ma'am, in this, because we are administering the radiotrobes in tracer quantity, so biodosimetry becomes a more important tool for assessing the toxicology or toxicity of that radio probe. So we are not injecting some milligram quantity of the radio probe. So we do the biodosimetry of different radioisotope. If I'm using technetium 99M, and according to radiation safety protocols and regulatory approvals, we see that this is the radiation burden to the mice when we inject 100 microcurie of the activity. And you see, we are injecting into only microcurie, sometimes nanocurie also. So very less activity is injected, and just to see where it has been, accumulated and distributed in the whole body. And accordingly, we do the assessment of using the software and the quantitation models. PMOD is a very nice tool to assess the tracer kinetics. We can do the 2-compartmental model and a lot of things in the tracer kinetics over there. But yes, toxicity, we talk in terms of the biodosimetry. How much doses we are giving to the rat or mice or any animal model. And you rightly said that if you're using the theranostic agent means, I have said that these are like simultaneously you're injecting the theranostic agent, or it's a sequential theranostics. They are two different things. So when the 10 half-lives of our radioisotopes is over, then we can work upon the second radioisotope. So this is what we do generally, because we calculate the PKPD, we do the PKPD, we do the pharmacokinetics, we know the blood clearance, and the plasma clearance also we assess. So after doing the plasma clearance and what is the plasma clearance rate and of the radioisotope, then we assess that the radioactivity is completely gone from the system, though it's not completely gone because half-life is, it depends upon the pharmacokinetics of the drug molecule that we have injected. So after the metabolism, we see that if the radioactivity is not there, the animal can be reused. We can reuse the animal, and all the studies that we do invasively could be done non-invasively, and it is acceptable also.
Okay, so there is one question here from Vaishnavi. She's asking, what are the possible mode of administration of those radio-labeled probes?
I mean, it entirely depends on the study that you are taking up. If you are, you need to see some, you have some intravenous injections, like you want to see do not pass first pass metabolism, and you want to do the IV administration, it is possible. Oral administration is also possible, and also nasal administration is also possible, wherein we do the lung imaging. We are giving the dose with the spray from the nose. So lung imaging may nose via nose route it is being given. So intratumoral injection is also given. Peritoneum also it's given. When we use the term scintigraphy, pharmacoscintigraphy, any kind of administration of these radio probes is possible, because you need to, even ocular route is also there, where you want to see that the eye drops, how they're working, if they're going in the systemic circulation or not. Then you label your ocular or the antibiotic, and then you can see ocular administration of that imaging probe, and you can see by pharmacoscintigraphy that it is going through the systemic circulation by using the dynamic spectral PET. So it is possible to use any kind of administration of these relevant probes. Yes, ma'am.
Oh, sorry, Andra, you can ask this question yourself.
Hello. Good evening, ma'am.
Good evening.
I have a question that what are the possible adverse effects of radiation when a children suffering from blood cancer is treated with radiation?
What are the adverse effects of the radiations? Radiation question in children suffering from blood cancer and treated with radiation. Although it's not her area, but she can always speak.
Yeah. So you can understand with this that the blood cancer, you're talking about the blood cancer.
She's asking about the children.
Yeah, children and blood cancer.
Both. Yeah.
Leukemia, that's a very, very, sensitive area, and because we always have to see the benefit to risk ratio. When the children, it's a last approach to, use the radiation on the children. Because if we give the whole body, dose to the children suffering from blood cancer, it could adversely affect the normal tissues also. Radiation could affect the radiation like the normal tissues also. So it should be the last resort when you're using the blood cancer and treated with radiation. So I think that in children, if the tumor is localized, and if then we are giving the PRRT based therapies, that is very successful in that. So this is what I feel, that peptide-based radiotherapy, and which is very, the radiation dose is very, very less. That I told the example of neuroendocrine tumor that has come out really well. So lutetium-based systems are there, lutetium-based peptides are there, that is being utilized to that can be given to the children that were suffering from the localized tumor.
So this is not effective against liquid tumors. The approach you are telling
approach is effective against that. But see, if I'm talking about the folate receptor imaging, or I'm talking about the proliferating cell imaging, so if I assess the number of receptors on oncological site and normal site, there is a difference of 20,000 is to 1, like 20,000 receptors in a generic manner, approximately 20,000 is to 1 is the ratio. So if I'm talking about that, and I'm talking about folate receptors, but folate receptors, they recycle back. They deliver 400 million of drug molecules in a very short span of time. They are overexpressing at the tumor site, even the LAT1 or any EGFR growth factor receptors, VEGF growth factor receptors, integrins receptors. These are overexpressed in a lot of cancer or the tumors. But if you're talking about the liquid leukemia or the blood cancer, you see that if you are injecting the directly into the IV, you are giving the IV, and it will definitely, it is going to kill the cancer cells, but it will also affect when you are giving it in the blood as such with the patient with leukemia, it will also affect the normal tissue also.
Thank you,
ma'am. Targeted, but yeah, it will be. You can again pose this question from Dr. Maria. She is a clinician and radiation oncologist. So maybe you'll get a definitely satisfactory answer from her.
So you have another question from Vikas Maurya. He's asking, most of the drugs are cleared at reasonable rates from circulation by kidney. Means he's talking about the second pass metabolism. So is radiolabel probe shows any adverse effect on kidney? First of all, maybe you should, they are asking how it will be cleared, clearing rate, and then they are asking if they are doing through the kidney, are they going to affect the kidney also?
So answer to that question is because whenever we are designing a radio probe, if I talk about the fundamentals of designing a radio probe, first of all, we need to have a high affinity vector molecule that is in the nanomole to picomole affinity towards its receptors. So that is why we call it as targeted radio probe. Second thing is that we need to consider its rapid accumulation at the target site. At the same time, rapid clearance from the body. When I'm saying this rapid clearance from the body, the clearance is done from the liver, hepatobiliary routes, or kidneys are used as a route of excretion. Now, if they are if they cleared at a reasonable rate from kidneys, definitely the kidneys are at stake because they're clearing the radiation. But we need to see that whatever we inject, it is always we try to assess the radiation burden to the organs also. This is what is called biodosimetry. So according to the biodosimetry, we standardize our radiation dose to the animal, to the human, so that minimum damage is done on the kidneys. Second good example is that how we can minimize the kidneys or protect the kidneys. We can, first give the patient some peptide or octreotide, like octreotide or some protein. So it gets and bind to the kidneys. So when we give the therapy to that patient with octreotide, lutetium-177 or yttrium-90, so you can protect your kidneys by giving a cold peptide because you have blocked the kidneys or you can also give polyethylene glycol per se, that also could protect the kidneys. So yes, there is definitely because the clearance rate is high when we making a small probe, they definitely excrete from the kidneys, and we make sure that we do pharmacokinetic modulation in such a way that it goes rapidly, cleared from the kidneys and does not retain at the kidney. If it is going to retain on the radiation is retaining on the kidneys, then it's bad. Then we are not going to use that probe at all. So we need to do the pharmacokinetic modulation also with respect to kidneys. We have to see whenever we are designing the molecule, we also see that we are modulating in such a way when it clears from the body, it gives minimum radiation dose to the different organs also.
Is that Dr. S.K. Mishra wants to ask something? Dr. Mishra is our RSO, Radiation Safety Officer. Dr. Mishra, we are not able to hear you. You can write in the chat box. If you want, you can write in the chat box. Okay, I think it'll take time or there's some problem. So I have one last question for you. Dr. Mishra? Hello? Palak, is he muted or you can unmute him?
Ma'am, he is not muted. He's unmuted only.
He's saying, give me a minute. Okay, let us give him a minute. He's saying. So Pooja, I just want to ask, by the time he asks the question, maybe this is the last one, that is there these theranostic agents or these kind of agents are being used other than FDG or all these things in the other part of the world? They are using it regularly?
Yes, ma'am. They're using it. Lutetium, ma'am, हमारे यहाँ पे भी we are also using in India. Lutetium-177 is utilized. Rituximab labeled lutetium-177 lutetium. So they're using in Fortis. So they're doing the therapy with these molecules. Even the and neuroendocrine tumor give very, very good results with these kind of therapy. Even actinium-225 also, it's a very all over the world. They are even University of Pittsburgh. They're using in the children's hospital. They're using actinium. They're working on actinium-225. That is a therapeutic radioisotope. So worldwide also they're using very extensively using yttrium-90, lutetium-177, actinium-225, statin-211. And samarium-153, ma'am, it's a one example of simultaneous theranostic, which is used in bone palliation. Quadramet is the brand name of that. EDTMP, which is ethylene diamine tetraphosphonate molecule, which is labeled with samarium-153, is being given for bone palliation. It gives very good results in bone metastasis. It's a proven molecule for that matter. And there are a lot of other examples apart from PRT, also PSMA-based pharmaceutical that I have shown it to you. A lot of all of the like worldwide and also in our country also, gallium PSMA is utilized for prostate imaging. I've shown choline. Choline is not available everywhere because it requires a cyclotron at site. So F18-based molecules, they require, if you have a cyclotron, then you can make it and you can use it. But gallium is such a radioisotope which you can obtain from the generator. So it's a generator-based. Germanium-68 is the generator that you can take and elute gallium-68 and then label your peptide molecules and do the imaging. So these are available at different centers now because even we have done the workshop in 2013 and given the protocol for labeling with gallium-68. So a lot of center in India itself, they're using gallium-68 PSMA based molecule for prostate cancer. And they are now using lutetium-177 PSMA also for the treatment. Yttrium-90, though they are not using lutetium-177, I have several studies are there from India. So there are like these kind of therapies, theranostics that are being utilized in India as well. Worldwide, so they're doing in a very nice way.
Okay,
Yeah.
so yeah.
Can you hear, ma'am?
Yes, sure.
Okay. So Dr. Pooja was nicely explaining about how certain organs can be protected while injecting certain radiochemicals. So just I wanted to add one thing, just it came to my mind. So it's the same way we can protect thyroid gland also when we inject say radioiodine or something, then by giving Lugol's iodine. So that goes and block thyroid follicles. So it will protect damage of thyroid gland. So just I wanted to add it came to my mind.
Certainly.
Thanks.
Okay, so I think Dr. Pooja has done a wonderful job. And any one of you want to collaborate with her, you can collaborate with her. She is a chemist, she's a biologist, she's a now half clinician also. So, and Mass have a wonderful, facility in terms of that, because most of the time when we want to do the research, the problem comes in university system that half of the time we don't have the very good kind of facilities there. We don't have radiation facility, we don't have animal facility. So in that way, in Mass has and the director in Mass will be with us tomorrow morning at 10 o'clock, and so you will come to know more about it. And I would like to once again thank Pooja for I just asked her to. He's again asking a question. What are the key properties? Sure, तीन question आ गए ना. एक तो you can answer if you can do. But anyway, thanks a lot, Pooja. If you can answer this question, it's fine.
Sure.
What are the key properties of substance must have to act as theranostic agent?
Yes, ma'am. I can answer that. So this is what I'm when I heard like theragnostic was a a term which was coined. First of all, theragnostic, therapeutics and diagnostic. They just joined the word theragnostics, therapeutics and diagnostics. So key properties of a substance. Now, what is this substance? You're talking about radioisotope, or you're talking about the normal what do I call the pharmacophore. If I'm talking about the pharmacophore, so pharmacophore is a targeting agent. Whenever we are talking about the pharmacophore, it becomes a targeting agent. But without the radioisotope, if I'm saying without radioisotope, I'm working on some theranostic agent. Without radioisotope, if I'm using, then I have to give in the quantities to obtain a biological response from that pharmacophore. So that is a therapeutic agent, right? Then when I talk about a diagnostic agent, for diagnostic agent, we are using the tracer technique. We are using in tracer quantity in nanomolar to picomolar range, we are injecting inside the body, and we are telling that this is the diagnostic agent. So therapeutic agent is something which is giving you are injecting in some higher amount and which is giving the biological response. Diagnostic agent is already you are labeled it with radioisotope, and then you are injecting and you are getting the only you want the imaging and distribution, so that you are injecting only a tracer amount or tracer quantity of that particular radio imaging probe. But when we talk about theranostic, so that is why I have distributed into two things: simultaneous theranostics or sequential theranostic. Sequential theranostic, when I'm talking about the radioisotope that I'm using, I'm talking in terms of radioisotopes. So if I'm using a radioisotope which emits both gamma as well as beta, that is True in the case of 153 samarium, which emits gamma and beta at the same time. So it becomes a theranostic agent. So it becomes a property of the radioisotope and not the pharmacophore. So if I talk about the property of the radioisotope, that this becomes the radiodiagnostic or radiotheranostic agent or radioisotope, radiotheranostic isotope that I'll be using with my pharmacophore to be called as a radiotherapeutic or theranostic agent. Then that will be the what do you call a theranostic agent. When I come, when I am using a sequential theranostic means that I want to see my drug is working and going at the target site. Like at the tumor site, it is accumulating. At the target site, I have seen with the imaging probe. Now, if I change that into some cold gallium, there are few reports that I have undergone. Cold gallium, if you label cold gallium also, that is also, you know, killing the tumor at the site. But you have to inject in more quantity because it is non-radioactive. So it becomes sequential, where the counterpart, the theranostic counterpart, therapeutic counterpart of that theranostic agent is non-radioactive. One is for diagnosis, it is radioactive. For therapeutics, it is non-radioactive. Then also it can come under the category of sequential theranostic. So sequential theranostics take care of the radiometal ion, radioactive and radioactive counterpart, as well as radioactive and non-radioactive counterpart. So this is how you can differentiate between different theranostic agents that you can design, define in your own terminology also. Because this is, I think, this is my terminology when I'm saying that therapeutics, I'm using non-radioactive therapeutics and I'm combining with a radioactive tracer technique. So I think I'm able to give an answer to your question.
Yeah, log up question पूछ रहे हैं. So now you have one question. Dear ma'am, please explain the contrast agent bioavailability in the system at the time of diagnosis when performed during the imaging process. How much it is bioavailable? I think it's 100% bioavailable. Because you're checking in blood or what? No.
Yeah, maybe. Actually, he wants to ask when the, okay, this is done two ways. Contrast agent, if you're talking about imaging probe, then also it's a different, answer to this question. If you're talking about MRI, then it's a different answer. Because in MRI, we need to inject a whole lot of gadolinium contrast to get that image. And if you're talking about the radio imaging agent, and for SPECT and PET, how it is done in real time. How it is done in real time? When we put the subject in the scanner, we inject, for example, I'm taking an example of FDG. You inject a patient with FDG, but FDG, because it targets glycolysis, we have to keep the patient for 90 minutes in a, entirely dark room and without any activity. He is not allowed to, or he or she is not allowed to, move your hand, move here and there. Just back, relax, so that when you move your hand, your glycolysis, like it increases and the uptake in the hand will increase. So we don't want that. So for that matter, there are bioavailability depends on that also. That if it is in the case of FDG, and if you are moving, you are doing exercise, you will have a lot of accumulation in the muscles that you will going to see in the scan. So what we do, we immobilize the patient in a dark chamber, and after 90 minutes we take the image. So that after urination, first we ask the patient to go and urinate, and after that we put them onto the scanner, and then we get a very good signal to contrast ratio. So bioavailability in the system, these are different protocols for different radioisotopes. For carbon-11 methionine, if I talk about that, nothing is required. No fasting is required. Even FDG requires 8 hours of fasting. But in carbon-11 methionine, no fasting is required. It can be injected at the time, and then after 20 minutes, you can put the patient in the scanner. So the bioavailability of carbon-11 methionine, because its half-life is very less, it's only 20 minutes. So you don't want to miss any of the lesion in the patient body. So what we do, we put it in the scanner and we analyze the whole body also and the region of interest, like in brain. Brain also we see. So this is how you have to modulate, you have to design in such a way that the bioavailability in the system at the time of diagnosis remain in that quantity that it has to be on the target site, but it should not be there in the background. So background clearance we need and accumulation of the tumor site. So these two things we have to take care whenever we are performing diagnostic imaging. And bioavailability in therapeutics, like radiotheranostics agent, if you are talking about the therapeutics, it has to be because we are using long-lived radioisotope. Lutetium also has a 10-day half-life, I suppose, and it has. So we want that it should remain at the site for a longer period of time. It should remain bound to the longer period of time. So we need to switch on to the antibodies. They have a longer circulation time, so that could be a good option to do the immunotherapy with the radiotherapeutic agents.
Yes, so Amandeep, why don't you ask your last question yourself? I know the answer, but she will give it. No, Amandeep, are you here? You're writing, obviously you're here.
Yes, ma'am. Yes, ma'am.
So ask the question yourself, loudly.
Yeah. Hello, ma'am.
Give your introduction also.
Hello. Hello, ma'am. I'm from SCMM and I'm postdoctoral fellow here. Your talk was very interesting, actually. It was showing a lot of different diagnosis processes, the methods, how the patient are screened. So my question is, when the contrast agent is used, so what is the clearance rate? Means if that is injected, so how much harmful it is? Because after some time, if it is in the body, is there any side effects of that contrast agent within the patient body? So my question is, what is the excretion, means how that thing is clear out from the body after the diagnosis is done?
Yes.
Yes.
See, if you see whatever radio probes that we have utilized, or they have a very rapid clearance from the body.
Okay.
70 to 80% clearance. I am talking about in case of radioisotopes, which is injected in very tracer quantity. So 70 to 80% it goes off within the first initial phase of clearance. So extracted, if you say the extracted coefficient of the radioactive percentage that goes onto the pathological site is 1 or 2%, or maybe 4% at the max.
Okay.
And the real scenario, maybe in the bone imaging agent, it becomes 10%. But the background will be cleared very rapidly. We design in such a way that it should be clear from the body in a very in a stipulated time.
Yeah,
and
if you see, I want to know that, like in case of brain imaging, so there is a blood-brain barrier. So what is the penetration like of this contrast agents? How much penetration it goes inside, and what is exact the strategy to diagnose the patient in case of brain tumor? So
it's a very nice question. I am happy that I'm able to incite a lot of questions in you.
It was really very interesting talk, because it is actually the depth of the radiobiology when the patient is diagnosed with a different type of cancer. So how it is diagnosed, so that is the main methodology. So you have covered that part. So it was really an interesting talk.
Yeah, so I will clear your doubt about blood-brain barrier.
Yes, ma'am.
Very nicely, very nice question. Because most of the drug development that is being done for the brain, for therapeutics, I'm not talking about the radio probes, but therapeutics, they fail because of this blood-brain barrier. So whenever we, see that whether the drug will cross the blood-brain barrier or not, it becomes a question of matter of fact that all the diagnosis and therapeutics depend on the uptake in the brain. So to answer that question, so the radio probes that we have utilized, like carbon-11 methionine, so it overexpresses LAT1 receptors or the transporters. So it is also seen that it is also there in the lining or the blood-brain barrier, endothelial lining. They also express LAT1 transporters. So with this also we see that the drug that we are making for the brain, it should clear, it should passes the blood-brain barrier. So we design in such a way that there is a high ratio like endothelial cell, LAT1 receptors. So they take the drug or the radiotherapy theranostic agent from there inside the brain. Then LAT1 transporter takes it to its sites: high-grade glioma, low-grade glioma, meningioma, astrocytoma, and in a different manner. There is a variation in all these tumors, so that is why I have written that this is good for prognosis. This is good for delineation. This particular molecule is recurrence versus necrosis. You can find out because every drug has its own limitation and its advantages. So prognostication, when you are looking at the prognostication and delineation for the surgery, FET, there is no better molecule than fluorine label FET. There's a fluoroethyl tyrosine. Because carbon-11 methionine, though it has a very high specific activity, it is the most because it is, you can't differentiate between a real methionine and carbon-11 methionine. But it goes to inflammation also, carbon-11 methionine. And even at our center, we have done several patients, and there's a case study reported that it goes in tuberculoma also. Carbon-11 methionine goes to tuberculoma. That has not been reported worldwide. At our center, we have seen that it has the carbon-11 methionine is useful in detecting tuberculoma in the brain also. So these kind of studies come when when we do a lot of human subject on these radio tracers. And if you talk about FET, FET was also found very helpful in detecting Ewing sarcoma. If you read about Ewing sarcoma, that is the adolescent tumor that happens to an adolescent. So in that case, if you are able to detect it timely, that it is a very malignant and proliferating tumor, with this FET scan, I think a lot of surgeries could be stopped at the same time.
Thank you, ma'am.
You're most welcome.
Okay, so nice. Can I ask everybody to open your video? We can take a one photograph. Whosoever is here physically have not, I know very less people have been left, but still.
Oh,
okay.
Dr. Moy is also there.
And she's not asking any question. This is very unfair. I can see Dr. Kanta, you are here from first day. So, but you never asked, you didn't ask anything. Would you like to?
Yes, madam, but my students asked the questions.
Okay, good.
We are doing with animals and lymphocytes for the past 10 years.
You're dean at SRM.
Yes, Center for Environmental Nuclear Research, and all the people who are interacting with you have visited my place. And Dwarkanath sir is my good friend, and Prasanna, NCI, NIH, all are, even Vikram also has visited my place in 2014. And in 2016, we conducted an international conference on radiation. So I'm in this field, and like Dr. Pooja has done, that was my passion to do that, but unfortunately I could not carry out the work. So I have done it in a different way. And one other one is certain aspect of radiation biology is missed out. Maybe next time I will explain that to you. As,
Yeah, you are correct.
That's also clinical aspect, because we are doing on wound healing. We are making some plasters for the wound healing using the polymers, for which when we use for clinical application, we go for radiation. We use the facilities available in Indira Gandhi Atomic Research Center. We have a good collaboration, and I got a lot of funding from 2009 onwards. We got funding from BRNS and things.
Yeah, because I saw you from morning to evening.
Yes, madam. My students and my, unfortunately,
Sunday,
I couldn't, and
I really
couldn't
felt great that you were doing your work also, and then you were attending.
Yes, because we are coordinating a lot of activities.
And I can see Mukund also. So Mukund, you are a Ayurveda biology student? Can you unmute? You are from which place? Huh?
I'm Ayurveda biology student, ma'am.
Okay. Yeah, so I saw you very seriously sitting all the time. So how are you finding it? Is it good for you?
Yes, ma'am. Yes.
कुछ समझ में आ रहा है कि सब कुछ French लग रहा है?
we'll try. I'm trying to understand, ma'am, because we are in the BSc.
Yeah.
Knowledge.
So JNU has started Ayurveda biology. So these are all second year and third year students with us. Good. So thank you very much, Dr. Uma and Dr. Uma Kerry, Dr. Pooja. And so she has done a great job, and we can have a photograph of everybody. And thank you. And maybe if you find time, Dr. Pooja, please join us tomorrow also. And I'm sure now you'll get too many emails and too many questions to answer. So fine.
My
Thank you
pleasure,
very
ma'am. My
much.
pleasure.
And now I'll finish this session, and I'm not playing any more video because I think you people are not interested in that. So we rather should have a live interactions. And so tomorrow we will again convene the session at 10 a.m. and Vice Chancellor JNU will be joining us, and INMA's director and one of the SD Sharma from BARC, a very senior scientist, is joining. So please be with us by 9:50 because tomorrow we have to run it. We will at sharp at 10 a.m. So thank you very much and thanks a lot. And it was a wonderful session, Pooja. And thanks for giving us so much of time. I know
so much, ma'am.
you're very busy. Everybody is busy nowadays. So thanks a lot. Thanks everybody and all the participants also. Thank you very much. Thanks.
Thank you, Dr. Pooja. It was very, very wonderful and very informative session.
Thank you so much, ma'am. Thank you so much.
So now you have one more person who, so Pooja always is a very good presenter. So that's a good thing, and always is smiling. That's a great thing.
Thanks
So
to our
thank
teacher.
you very much.
Thanks to our teacher.
And
no?
being in INMAS and being in so much of, say, being a woman, being a, say, mother, and then in between so many, great scientist of DRDO, she survived and she has come up so up, and I wish you all the best in your life. Thank you
so much.
Bye bye.
Thank
Bye
you,
bye.
ma'am.
Bye bye everybody. Thanks.
Bye, ma'am. Thank you. Bye, Pooja.
Bye.